AlZayer. I'm a third-year medical student, and today I'll be teaching you guys gastric mucosal defense 1 and 2. Okay, so you guys can still see my screen, right?
Yes.
Okay, perfect. Okay, so these are all of the objectives we'll be covering. We'll be covering gastric mucosal integrity, the 3 layers of the gastrointestinal mucosal barrier, prostaglandins, peptic ulcer disease, hiatal hernia, H. pylori, NSAIDs, Zollinger-Ellison syndrome, vomiting, and at the end we'll have like a small pop quiz practice questions where like, you know, I'm gonna ask you guys questions. Okay, first I want to start whenever you guys have questions, write them in chat, and all throughout, like, I'll have the chat open and I'll check your questions all throughout, like, as we go. Okay, does that sound good?
Yep, sounds good.
Okay. Also, like, I feel like with unmuting yourself, I might not like be able to hear you clearly, and all of like your voices will clash. So anything you have to say, like, write it in the chat, and I'll always read the chat. Okay. So first, I feel like I'm going to start with a small recap to get you guys like in the zone, you know, for this, like, because to understand this lecture, I feel like you need a bit of basics beforehand. So you guys can write your answers in the chat. But first, I want to ask you, what is the digestive function of gastric juice? Write your answers in the chat. Yeah, okay, perfect. Yeah, okay, so the digestive function of gastric juice is hydrolysis of ingested proteins. Means it breaks down like proteins, that's the basic meaning. And it also has a bactericidal effect. Bactericidal means like it kills bacteria, basically. So what does oxygen get secreted with? Write the answers in the chat. Okay, I don't know. Okay, I don't know if you guys know. Okay, but pepsinogen gets secreted with HCl, which is like acid. And yeah, what's the role of HCl? The acid, what's the role? Do you guys know the role of the acid in the stomach? If you don't know, like, just say I don't know, because like I don't want to like wait and then yeah, perfect. Said no. Okay, so the main role of HCl, which is acid, is autolytic conversion of pepsinogen to pepsin. So yeah, it just converts pepsinogen to pepsin, and that activates it. And what's the role of pepsin? Yeah, perfect. So the main role of pepsin is basically to digest protein. So the release of HCl and pepsin is what makes the stomach very acidic. This makes the stomach's like pH level around 1.5 to 3.5. Okay, so moving on, I added this slide. By the way, I'll be sharing my presentation with you guys later, so you guys can like go over, and I transcribed everything for you to make it like easier on you. And also I'll believe that the YouTube recording will be uploaded, so yeah. But I added this slide for you guys. This is a slide from Dr. Peter's book, and I really advise you guys to read Dr. Peter's book, or like just go through it, because I mean, like, it's Dr. Peter who like worked on the book, and he's the one that's writing your questions. So yeah. And this is like the physiology and pathophysiology of digestion. That's the name of the book. And this slide just for your further understanding. Yeah, he has a book, and I'll yeah, you can read this on your own time. It's basically the function of gastric acid and pepsin. Okay, so now the question is, how does the stomach or the gastric mucosa protect itself from its own digestive secretion? You know how we said that acid, like acid and pepsin, all of that gets like secreted in the stomach, making the stomach pH level 1.5 to 3.5. We just mentioned this, right? But we also know that acid can harm the mucosal, the mucosa, sorry, the lining of the stomach. So how does the like stomach protect itself from that? Does anyone know? Write your answer in the chat. Yeah, perfect. It secretes bicarb and mucus, along many other things, and that's basically what we'll be covering in this lecture. So the answer is a mucosal defense system is in place that serves to prevent or at least minimize mucosal injury. And that's the entire thing that we'll be covering in the lecture. So what is gastric mucosal integrity? So as we just said, that we have like acid and pepsin in the stomach, but the stomach itself also needs to protect itself from this gastric and pepsin. So we have damaging forces, which is the acid and pepsin, but we also have defensive forces that the stomach uses to protect itself. So the gastric mucosal barrier constitutes of, and all of this, by the way, the defensive forces, this is called the gastric mucosal barrier. It's the barrier that the stomach uses to protect itself. And it constitutes basically of mucus, bicarbonate, restitution, blood flow, and prostaglandin. And this is what we'll be focusing on in the lecture today. So moving on. So this itself, if you guys can see the slide, this is the gastric mucosal barrier. It constitutes of 3 layers. Can I repeat? I'll be discussing this. So basically what I said is that the stomach protects itself using the gastric mucosal barrier, which is the entire lecture. So I didn't really explain anything yet. That's what I'm going to be explaining now. Okay, so this is the gastric and mucosal barrier. Okay, and it's stratified levels of protection. What does this mean? It has 3 levels of protection. Why do we have these 3 levels of layers of protection? To prevent mucosal injury, inflammation, and associated sequelae. What does this mean? What does sequelae mean? It's the result of a condition which is the consequence of a previous disease or injury. Okay, this might be a bit confusing. So let's say, for example, like I have a cut, right? The initial injury is the cut. And let's say like I got bacterial infection, this is the result. This is only an example for you to understand what the meaning is. So back to this, we have 3 layers of protection from the acidic stomach. The 3 layers are the pre-epithelial layer here, and the epithelial layer and the subepithelial layer. You can think of it this way. Let's say there's like a king here, okay, at the bottom. And there are 3 lines of soldiers, which are these 3 lines of soldiers that need to get to the king, that protect the king from like the enemies. Let's say the stomach acid is the enemies, which is here in the stomach lumen. Okay, and if you pass one, like for example, let's say the enemies pass this layer, the first line, they'll always have the layers under it to protect it from reaching the king. Okay, so you only lose protection when you pass all 3 layers. Okay, so the first layer, so basically what this means, the king example, is that we have a stomach that's very acidic. Okay, we need to pass all of these layers to get to the stomach gastric mucosa, okay, for it to be injured. And these 3 layers, they protect it. So let's first I'm going to talk about the pre-epithelial layer, and I'll go in further detail. I'm just going to give you the what is it called? Thanks, Ragda. Okay, so the pre-epithelium, this is the pre-epithelium. It's in the lumen. Okay, you learned this in your last lecture, I believe, about the alkaline bicarbonate mucosal. So this is what makes like the bicarbonates and mucus. These 2 things are what make the pre-epithelial layer. Okay, the mucus in the bicarbonate, in the pre-epithelial layer, it's very viscous. You can think of it like honey. Things are very hard to pass through honey. So you can think of the mucus in the pre-epithelial layer that protects it. Like it's very hard to pass through. And yeah, okay. So basically what the mucus does is that it limits the penetration of ingested microbes. For example, like bacteria and all the toxic like material, that limits the penetration. The mucus limits the penetration of all the things that we don't want, okay? We also have bicarbonate. So we said the pre-epithelial layer constitutes of mucus and bicarbonate, and we discussed mucus, and now we'll be discussing bicarbonate. Bicarbonate is considered a chemical barrier that neutralizes the acidic environment. Okay, so since it's alkaline, we just said that the stomach has HCl and like pepsin and all of that. That makes the stomach very acidic, of a pH like around 2, as you see here. And we have bicarbonate, which is an alkaline, and this neutralizes the acidity so that it doesn't harm the gastric mucosa, okay? And this prevents epithelial acidification. So the second layer, which is the epithelial layer, I think this one, okay? This is a single layer of epithelial cells that have tight junctions. Do you see the black dots here? These are tight junctions, okay? And what are these, like, how you can think of it? You know how we said like 3 layers of like soldiers? I'm trying to like help you guys remember it. These tight junctions, you can think of them as the shields that the soldiers hold. Let's say this is 1 soldier, 2nd soldier, 3rd soldier. These are the shields, okay? So what is like, they're in between each cell, okay? And they add additional protection. It's like, it makes it harder for the microbes from this, let's say like the microbes pass here, it makes it harder for them to pass. This layer also has a self repair mechanism. So if an injury occurs, it can repair itself. The 3rd layer is the subepithelial layer. And going back to our like enemy's king thing, if the enemies manage to pass both of these layers, the pre-epithelial and the epithelial layer, they reach the circulation, which is the 3rd layer, which is the subepithelial layer. And this acts basically as the last layer of protection to remove any noxious material. Okay, so
can I ask you a question?
Yeah, sure.
In the epithelial layer, it acts like a physical barrier. So like it's basically like it acts like a physical barrier, like between, we have cells and the cells are connected through a tight junction. So these cells prevent anything from penetrating the epithelial layer? Or
Yeah, this is like basically a layer of protection, okay? And it not necessarily prevents the like epithelial layer, it prevents the, like, you know how like the stomach is very acid, it prevents the stomach from it prevents the acid from harming the stomach itself, because like we don't want acidity, if that makes sense.
Okay.
Okay.
Yes.
And we will explain it further. Like, this is just an explanation. This is just like a introduction. Does the epithelial layer secrete mucus in the pre-epithelial layer? We'll discuss this now. We'll discuss each layer alone now. Okay, so let's look at each layer alone. First, we'll be discussing the pre-epithelial layer. Okay, so this is the pre-epithelial layer. Okay, remember here, do you see this, where is it, back to this slide, do you see this picture? You know how we said there are 3 layers, the pre-epithelial layer, the epithelial, and the subepithelial. Now I'll be discussing this layer alone, the blue layer, the pre-epithelial layer, okay? So the first layer of protection is the pre-epithelial layer, which is composed of 2 functional, okay, not anatomical, functional layers, okay? The outer mucosal layer and the inner mucosal layer, okay? Do you see here, this is the outer and the inner, okay? What do I mean by functional, not anatomical? Like, they're not actual, like there isn't a line that separates them. It's just they're functional for like functional purposes. Okay. So now I'm going to be talking about the mucus layer. Do you see here the outer layer? Where is it? The outer layer and the mucus layer, the inner layer. I'll be talking about the outer layer. Okay, this layer. So the outer layer, which is the mucus layer, it's a physical barrier because it's mucus. Like, you remember the honey analogy? It's mucus. The honey is like very viscous, and this is a physical thing. So the mucus is a physical thing. You can touch it. This is acts as a physical barrier. So as we mentioned, it's composed of 2 layers. An outer, so the outer mucosal layer, which is a physical barrier, and an inner bicarbonate layer, which is the chemical barrier. So now we'll be discussing only the mucus layers I mentioned. There are many names: mucus layer, outer layer, or the physical barrier. Okay, this layer itself is composed of 2 layers. So I don't want to get you confused. Okay, here I said that there is an outer layer, right? Outer and inner layer. This mucus layer, do you see this mucus layer itself? This layer is composed of 2 layers within it, if that makes sense. Okay. what are these two layers of the mucus layer? It's the inner mucus layer and the outer mucus layer. So the inner mucus layer is very dense and very viscous. Do you remember the honey? Remember that for the inner? Did you see this? Okay, this is the inner, adherent, firm. These are also names of the mucus layer. Okay, it's a very dense. Do you see how the green lines are very thick? It's very dense and viscous. I put the wording here in brown for you to remember that it's brown like honey. So it's very viscous, okay? This layer is adherent to the surface, and this layer, since it's very dense and viscous, first, it does not let a lot of bacteria pass through, and it's not removed by motility. Okay, so you know how the stomach, I'll be explaining it like further now, but you know how the stomach like has motility to like move on the food you eat, okay? This layer doesn't get, this layer is adherent, okay? It doesn't get removed by motility, okay? This inner adherent layer also gives rise to the outer layer. This is the outer layer, outer loose layer. This layer here and the inner layer gives rise to the outer layer. Okay. Now I'm going to be talking about the bicarbonate layer. Okay, but first, wait, before we do that, I just want to mention something. The outer layer, okay, it contains bacteria, and it's removed by motility. So you know how there's three layers. Let's say that we have an acidic stomach. Okay, and we know that acid is a toxin. Okay, it manages to pass through the or let's use bacteria. I feel like since bacteria is the one used here, let's say there's bacteria here, okay? Bacteria is also gonna be in the loose mucus layer. Since it's very loose, bacteria can like pass through. It's not gonna be as much as in the lumen itself, but you'll find bacteria in the loose mucus layer, okay? Here, since the inner layer is very dense, okay, it's very hard for the bacteria to pass through. Also, you know how I said this dense layer, since it's very tight and it's very like attached to this layer, okay? It will not be removed by motility, but this layer, since it's loose, with motility, it can be removed. And why is this a good thing? Because we want all of this bacteria to be removed along, since we said that like we don't want bacteria, right? Okay, so, or any noxious material in general. I just use bacteria for an example, okay? We said that this layer contains a bit of like bacteria. With motility, this layer will be removed. Okay. So as we slowly move away from the surface epithelium, you see here, this is the surface epithelium. As we slowly move away, the density decreases. Do you see how like here it's very tight and here it's not? It isn't as strong, so it forms this layer. So the inner layer, or the adherent or firm layer, as it moves along like upwards, as you can see here and here and here, it becomes looser. Yeah, that's what I'm doing right now. I'm repeating. So the mucus layer in general, it's composed of two layers, the inner and the outer. The inner one is very dense and it's very compact. It's viscous and it's attached to the surface layer here. Okay. As we move upwards, the density decreases, okay? As you can see here, they're not, this layer isn't as tight as this layer. See, the green here is very thick and the green here isn't as thick, okay? So this layer, the inner adherent layer, as we move upwards, it becomes less dense, and then we make this layer. So this layer and the inner adherent layer makes the outer loose layer. So you may be asking yourself, how does this happen, and why does the dense mucus layer make the loose layer? Like, why does it become looser? It becomes looser due to proteolytic cleavage using glycoprotein, which is pepsin. So pepsin, okay, through proteolytic cleavage, makes this like dense layer, as it moves up, looser and looser to make the outer loose mucus layer. Okay. And the outer mucus layer, it contains bacteria, but bacteria doesn't easily penetrate into the inner layer because it's, as we said, it's thick and dense, okay, and very viscous. And the outer loose layer gets removed and renewed. Why do we do that, or how? Okay, it's by something called the BER. Now, what's the BER? The BER is the basoelectrical rhythm. Okay, remember the motility that I was talking about min shway when I said that through like stomach motility, this layer will be moved out. The motility is the BER. Okay, this is the motility that occurs 3 times a minute that empties the outer layer into the small intestine. Okay, and gets rid of it. So this layer through the BER, which is motility, every 3 times a minute, I think. What was it? Yeah, 3 times a minute. Okay, will empty itself or renew. Okay, and it will empty all this material into the small intestine, which is after the stomach. And why is this a good thing? This is a good thing because we want to get rid of all the noxious material and all the bacteria and microbes that we said were in this layer. You see the black dots here. This is all the bacteria and noxious material. And we want to get rid of that because we don't want that. Okay, is that clear for the mucus layer? If it's clear, right? Yes in the chat. Okay, perfect. Okay, now I'm going to be talking about the bicarbonate layer. So going back, we said that, I want to go take you back to the first image. The pre-epithelial layer, okay, the first one, okay? This is made up of a mucus layer and the inner layer, or the outer layer and the inner layer, the mucus layer and the bicarbonate layer. We discussed the mucus layer, now I'm going to be discussing the bicarbonate layer. What makes the inner layer, the inner mucus layer? It's made up of mucus. I don't understand your question. It's just in it. It's secreted by, oh, what secretes it? It secretes it like in the stomach. It's by, I think there's some cells, to be honest, I wasn't the one that taught you the secretions, but there's special cells. I think like goblet cells in the stomach.
Mucus cells?
Yeah, there's like specific cells in the stomach, okay, mucus cells or like goblet cells, I think they're the ones called in the stomach that secrete mucus that make up this layer. I'm gonna, search it for you eventually and write it. Also, if you have like further questions that maybe I'm not able to answer, like you can always email me or text me, and I'll send you, my email is included in the first slide and I can send you my number later. Okay. So now I'm going to be talking about the bicarbonate layer. The bicarbonate layer is the chemical barrier. Okay, so this bicarbonate layer, as we mentioned before, creates a pH gradient. Okay, what does this mean? It makes the surface epithelium at a pH of 7. Okay, and the stomach lumen at a pH of 2. Okay, so do you see the lumen here? This is the stomach lumen. This is a pH of around 2, 1.5 to 3.5, well, like around 2, okay? This is the outer mucus layer, the one that we talked about before, okay? And here, this is the inner layer composed of bicarbonate, okay? This is a chemical barrier. Why is it a chemical barrier? Because it's it's alkaline basically, and it stabilizes or neutralizes the pH, okay? So we mentioned this concept before. I talked about it before. Is everything clear so far about the mucus layer and the bicarbonate layer? Okay. So also, this is the mechanism of bicarbonate secretions into mucus. This has been covered in like your previous lectures. So I just wanted to include it for to keep it in your mind. And yeah, the outer mucus has an outer and inner layer, and the bicarbonate is just a bicarbonate layer. Yes. Okay, so before we move on to the second layer of mechanism, which is the epithelium, let me just quickly revise this layer, okay? This is the pre-epithelial layer. It's composed of the mucus layer and the bicarbonate layer. The mucus layer itself is composed of the inner with adherent firm layer, okay, which is the very dense one, and the outer loose mucus layer. And then there's the bicarbonate layer, which is the chemical barrier. And the mucus layer itself, composed of the 2 layers in it, is a physical barrier. Clear on that? Did I answer your question, Siham? Okay. So we finished discussing the first line of soldiers, you can say, or the first protective layer, which is the pre-epithelial layer. Now I'm going to be talking about the epithelial layer. So let's say the epithelial layer is breached. Breach means it's broken. So let's say bacteria or noxious material managed to break through the first pre-epithelial layer, the one that we just talked about. The next thing in line, the next like soldiers that have to stand up and protect, okay, is the what is it called? Is the epithelial layer. Do you think this session is enough, or should I go back to the doctor's lecture? Personally, okay, I worked so hard trying to include everything from in this like small PAL session, but I always do encourage you to go back to the doctor's lecture. Maybe he emphasized on a point or something. I encourage you to like skim through it. Also, if you have any questions, you can always like ask me, like in this session, outside, through my email or like WhatsApp or something. Anywho, okay, so as I just mentioned, if the pre-epithelial layer is breached or broken, the next protective line, okay, is the second line of soldiers, and this is the epithelial layer. This is the one that I'm going to be talking about, okay? There are two very important points in the epithelial layer, okay? The first is the tight junctions. Here, the ones that I wrote in pink, the black dots, this is the tight junctions. These prevent paracellular efflux. Now you might be thinking, what is paracellular efflux, okay? This means, okay, that it prevents the ingested material to go in between the cells, okay? So let's say there's like bacteria here. These tight junctions, you remember when we said they're like shields, it adds extra protection from any like noxious material to pass through. Okay, this is basically what paracellular efflux means. Okay, and yeah. So if these tight junctions break, the noxious material will be able to enter in the interstitium, causing two things. Okay, and I wrote them in red. The two things that can happen if the tight junctions are like broken, okay, or like don't work. Okay, it will elicit a local inflammatory reaction. Okay, how does this happen? Because it will activate the immune cells. Okay, in the interstitium here, okay, there are immune cells. Okay, if they get activated, this causes inflammation. Okay, so let's say there's bacteria here, the tight junctions aren't working, the bacteria passes through the epithelial layer. Okay, it will end up in the interstitial fluid, in the interstitium. Okay, what is in the interstitium? Immune cells. What do these immune cells do when they get in contact with bacteria? They cause a local inflammatory reaction because they will get activated. Okay, and yeah, it's basically part of the protection. Okay, also, if the inflammation is so severe to the point where it's like, yeah, so severe, it can spread throughout your body, causing systemic consequences such as sepsis. What's sepsis? It's basically bacteria entering the systemic circulation of your body. Okay, so to quickly recap, okay, the epithelial layer is constituted of two main important points: tight junctions and restitution. What I just talked about are the tight junctions. These are the black dots here, and they prevent paracellular efflux. That's the mechanism. Okay, they're like soldiers. What happens if the tight junctions aren't working? You can have two main things. Okay, it elicits a local inflammatory reaction. Why? Because the bacteria can pass through and get to the interstitial fluid with immune cells in it, okay, and they will react, causing an inflammatory reaction. Second, if the inflammatory reaction is so severe, okay, it can cause a systemic consequences such as sepsis. Okay, and the second thing about this, the important thing about this, second important thing about this layer is restitution. Okay, restitution, we already kind of like discussed it before. It's means repairing of the epithelium. Okay, that has been injured. So this layer has a mechanism that can, like the epithelium can get repaired if it gets injured. Okay, how does this happen? What's restitution? Restitution is epithelial, like the repair mechanism, okay? So once an injury occurs that damages the epithelial cells, the epithelial layer, okay? If it damages them enough for them to detach and undergo cell death, okay, let's say here, do you see this image? Okay, let's say injury occurs, okay? Injury, these cells get injured, okay? It gets injured severely to the point where the cells detach and cell death occurs. What happens? Epithelial cells next to the injured part. You see, this is the one like in this purpley color, okay? These are the injured cells that undergo cell death, okay? After they detach, cells next to them, the white healthy cells, okay, they will start to change shape and migrate to cover the injured part. Do you see the small like squiggly lines here? Okay, this is when these white cells here, the white colored cells, these are the healthy cells, okay? They will change shape and migrate to cover this part, okay? Because now this part is exposed and we don't want it to be exposed because we want to protect it, right? So these cells, they migrate and change shape and they try to cover as much area as possible, okay? This new layer of cells that form, by the way, do you see this? This is the small like cells in the middle, okay? This is what gets formed, okay? What is this called? This is called a monolayer, okay? Monolayer of cells. These cells are not columnar as the normal cells next to them. Do you see these cells? These are columnar in shape, okay? But these cells, they're not columnar, the ones that migrate and change shape to form a monolayer, okay? This all happens very, very fast. So all of these steps like injury and then like after cells like die and detach, okay? The mechanism of restitution, which is these cells, the white cells next to them, they migrate and change shape and form a monolayer. All of this happens very fast, okay? And why does it happen very fast? Because we want to heal it very fast. We want to heal it as like fast as possible for it not to worsen. So it takes around like minutes to hours. Okay, how can you think of it? How can you like memorize this? You can think of it as a group of friends, okay, of 3 people. Let's say there's a group of friends of 3 people. 2 groups of friends, let's say this one, they're fighting, okay, and they're drifting apart. But the 3rd friend tries to remain their bond. So let's say like this is like the 3rd friend is trying to like extend a hand from this side and a hand from this side, trying to like pull the 2 other friends together, okay? And you know, like this friend in the middle got squished, they're trying to like hold the cells on the side together, you know, okay, to remain the bond. But the 3rd friend is stretching so much, like it's stretching. Do you see how it's stretching? It's not like the same level as the other one. The other ones are columnar. Well, this is a monolayer. It's not like columnar. Okay. Yeah. Okay. But like as much as like the friend is trying so hard, the cells themselves, they have to like, I'll get to that eventually, but just think of it like that. Like there's a 3rd friend trying to like maintain the bond between the other 2 friends. Okay. The important thing to note is that these cells are not undergoing replication, okay? So do you see this monolayer? This is not replication. This is just migrating. These cells are migrating and they're flattening out to cover as much surface area as possible. So this entire like process, the restitution process, okay, this is only temporary, okay? As these cells or these cells, the 3 cells here, the monolayer, okay, it only aids in helping the situation for some time. Like complete healing is required, like that makes sense, okay? And that's the difference between restitution and complete healing, okay? Do you remember the example that I just gave you? The 3rd friend is trying so hard to maintain the bond between the other 2 friends, right? But like the 3rd friend that's holding the bonds can't do that forever. The 2 other friends, they have to heal their bond, or they have to heal their friendship like on their own for the long run, if that makes sense, okay? So, and that leads us to complete healing. Okay, this is the process of complete healing, okay? And I'll explain it now, of course. And this process takes around 3 to 4 days. So we said this process, the restitution process of like temporary, this is very temporary and it takes minutes to hours, okay? But it cannot maintain like this forever. Complete healing needs to occur, and complete healing takes 3 to 4 days, okay? So what happens in complete healing? Stem cells divide and migrate and eventually differentiate. Okay, so let's say this is a normal like cell layer, okay? Part of the cells undergo cell death, okay? And they detach, okay? Now this is where restitution comes, the one where this example, the one that we gave, okay? This is when restitution comes in and forms this temporary, you can call it so-called Band-Aid. I think Dr. Peter referred to it as Band-Aid, okay? Yeah, it's basically this monolayer that's temporary, okay? But complete healing, it needs to like happen for this cell layer to like regain its full function, okay? This restitution part takes minutes to hours, while the complete healing takes 3 to 4 days. How does this complete healing occur? Stem cells, they migrate and eventually differentiate to make the cells normal again, okay? Does that make sense? If it makes sense, like, please write yes in the chat so like I know. Okay, perfect. Okay. I think can you guys still see my screen?
Yeah.
Okay. I don't know my screen once. Okay. Yeah. Replication and restitution. Do you see here? Do you see this monolayer that gets formed? The cells that are like shorter than the columnar cells next to it. This is the monolayer. Okay, this layer itself, these are cells, these aren't replicating. These cells here aren't replicating. They're just migrating, which is they're traveling, and they change shape to form like a small monolayer. Yeah, these cells change shape to form restitution. Okay, the temporary healing, while the complete healing, okay, this is when stem cells they replicate. Like this is when stem cells migrate and eventually differentiate. Okay. Yeah, for complete healing there is like replication, but this is through the mechanism of like stem cells. You guys took stem cells, right? Yeah, that's basically stem cells. Stem cells are like the most basic type of like cells, and then they migrate and differentiate into like the different types of cells in an epithelial layer, or like any cell layer. Okay, makes sense? Do I move on? So they migrate to the place, then replicate, differentiate. The stem cells, yeah, the stem cells in complete healing, they migrate to the place where there's like cell death, where healing is required, and they differentiate into the cells that are in that specific layer. If it's like parietal cells, if it's like goblet cells, whatever cells it is, they differentiate into that cell, the one that like got injured. It basically replaces the old cell that got injured. Has the same mechanism of the old cell that gets injured. Does that make sense? To like regain full function. Clear? No, in restitution healing, there isn't replication. In restitution healing, it's the cells themselves. Do you see the cells next to them, the columnar cells? This is what forms the monolayer. The cells next to them, not the stem cells. This monolayer isn't made up of stem cells. This is made up of the same cells itself that makes the epithelium. Okay, I'm so sorry if I'm going really fast. Okay, I'll try to slow down. Let me re-explain this. Okay, let's say this is the cell layer. Okay, can you guys see my hand? I think you guys can see my hand. Okay, so this is the cell layer. Okay, and let's say in between, so this what is it called? The blank space in between now. This is cells that have died. Okay, this cell layer, by the way, this is the epithelium. Let's say of the stomach mucosa. Okay, let's say it's made up of like parietal cells, any type of cells like you can think of in the stomach. Okay, let's use parietal cells for example. Okay, let's say this is parietal cells. Let's say the parietal cells in the middle, they died and they detached, okay, forming this space, okay? This is still parietal cells and this is still parietal cells. So the parietal cells next to them are still healthy, okay? The cells next to them are still healthy. So what happens is that these cells, they migrate, okay, and form this monolayer. Do you see how they migrate here? So it's not stem cells. These aren't stem cells. These are the actual cells that make up the epithelial layer. Okay, they migrate and they change shape and they form this monolayer, the monolayer here. Okay, where the stem cells come in, the stem cells are for complete healing. So do you see the monolayer, this one? The monolayer here, they're made up of the cells next to them. Let's say these are parietal cells. These are still parietal cells, but these parietal cells are like, they change shape and they migrate to just form this small monolayer, okay? Just like it's like a band-aid. Like I cut myself and I stick a band-aid temporarily until like it fully heals. This is basically what it is, okay? So do you see this? Let's say these cells died, okay? The cells next to them, do you see how they are slowly changing shape and like forming like a small band-aid, like a small monolayer? This is what restitution is. And these are not stem cells. Stem cells are here in complete healing. Here, it's not stem cells, okay? This is just for like temporary protection. This is what takes minutes to hours, but eventually this can't last forever. Like this will not help us, like, and this will not protect us forever. And this will not, these cells, they will not have like normal function. We need to get back the old cells here, the old same cells here. We need to get them back, okay, to have normal function. And that's what happens. Stem cells, they differentiate into the same cells that were here, the old cells, and they form new ones, but with the same function as the old one. Yes, restitution is just a temporary fix, but we need complete healing. We need like complete, not temporary, like permanent fixture, okay? And this is what complete healing is. Does that make sense so far? Okay, I thought you guys are calling me doctor, like call me Meral. Okay. Anywho, so now we'll be moving on. So like so far restitution and everything about the epithelial layer, restitution, complete healing is clear. I can move on to the subepithelial layer. Right. Okay, perfect. Okay, so now I'm going to be moving on to the subepithelial layer. Let's just go back for me to show you what layer to orient you. Okay, no, I don't want this picture. I want this picture. Okay, so we discussed the pre-epithelial layer, we discussed the epithelial layer, and now we're going to discuss this layer, the subepithelial layer. If you study from my PowerPoint after the doctor, would that be fine? I included like everything. I think you asked me the same question before. I included everything like I tried to include everything the doctor included. As you can tell, by the way, I included so much of the doctor's like own pictures and slide because I thought it was like they were good for like explanation. I think like, yeah, but I feel like I don't want to I don't want to like just say yes and then like miss anything by accident. So I still advise you to go through the doctor's lecture, to just like skim through it to make sure like you didn't miss any points. As I also mentioned, like I advise you to look through his book. I included the name in one of the slides. Thank you for saying that my slides are murattabin. I tried so hard. Yeah, I also transcribed my session. Like everything I'm saying, I included it in the notes section of this PowerPoint. I'll be sending it to you guys later. Okay, so let's move on so we don't waste time. Okay, so now I'm going to be talking about the subepithelial layer. So now let's go back. Let's say that noxious material managed to break through the pre-epithelial layer and all of its components. We said the components of the pre-epithelial layer are the mucus and bicarbonate layer. Let's say the bad material managed to break through this layer and managed to break through the epithelial layer, which is the second line of soldiers, okay? And all the components, which is the tight junctions and like all the restitution, complete healing. Let's say it managed to break through both of these layers. Which layer will it reach now? It will reach the third line of soldiers. Okay, remember the three line of soldiers, the pre-epithelial, epithelial, and subepithelial. It will reach the third line of soldiers, which are the subepithelial layer. So let's say it breaks through the first line of soldiers, the second line of soldiers, and it will reach the third line of soldiers before it can actually get to the king himself, okay? So what's the third line of soldiers? It's the subepithelial layer. So yeah, this layer is like very fast. So it's saying it's finally my turn. So after the material ends up in the interstitium here, okay? Now it's the job of the circulation to protect the gastric mucosa by washing out and neutralizing the acid. So let's say there's like bacteria and all the noxious material that came into your body, okay, in the stomach lumen, okay? And let's say the acidity itself couldn't like get rid of it. It passes through the mucosal layer, which is composed, or the pre-epithelial layer, sorry, which is composed of mucus and bicarb. It also manages to pass through the epithelial layer and its tight junctions, and this the tight junctions which are the shields. They were not enough to protect. Now comes in the third line of defense, okay, which is the what is it called? The subepithelial layer. And what is the subepithelial layer? It's basically circulation. Okay, it's the capillary. Okay, which like here, do you see the capillaries? Is basically the circulation. Okay, which will protect the gastric mucosa by washing out and neutralizing the acid. Okay, so how does it do that? For example, like here we have let's say like acid, and we don't want acid to get to the stomach, right? So let's say acid manages to pass through here and pass through here. The circulation will like wash out all the noxious material and bacteria and also neutralize the acid. Okay. I believe that like this slide, the doctor included in his slides, so I also decided to include it here. But I believe that you took this in previous lectures. Okay, I'm just gonna like quickly recap it. If you have like further questions on like, if you can tell, like I included like additional slides throughout my presentation just for you to like look back on. Either they were covered like previously in other lectures, and I'm sure like other tutors covered them, or some of the slides I add them myself, like for you to like further reading and understanding. If there's anything you don't understand from those, okay, you can email me and like text me. Okay, I'll try my best to like answer you over email and text. Okay, so as I said, like you took this in like previous lectures. Basically, remember when we said in the recap slide when what does acid get secreted with? Doesn't that cause acidosis? What causes acidosis? Going to the capillary. No. It's not. Okay. Neutralizing, what I meant by it neutralizes the acid itself doesn't get into the lumen. It doesn't get into the capillary, sorry. I will explain now how the capillary layer or the circulation helps in like neutralizing the acid. That's what I'm going to explain now, okay? Okay, so when acid gets secreted into the lumen, okay, do you see here? Acid, which is acid flow, it gets secreted into the lumen, okay? Through like these cells or whatever. Okay, this is the epithelial layer, okay? And you know that parietal cells and whatever, chief cells and all your other cells, you took that in other lectures, okay? They secrete acid, which is HCl. They secrete it here in the lumen, okay? What else gets secreted? Bicarbonate gets secreted into the blood, okay? Not in the lumen, as much as like all of these get secreted. Yes, we will have, don't get confused, we will have bicarbonate, okay? But with HCl, when it gets secreted into the lumen, at the same time, bicarb will get secreted into the blood, okay? Later on, this the bicarbonate that's in the blood, okay, it will go through the transporter. Like let's say bicarbonate gets secreted here, it will go up in the blood, and it will get secreted through the transporter, the bicarb transported, okay, and it will get secreted here into the lumen, okay? And this is what forms the bicarbonate layer. Remember when we said the first pre-epithelial layer, it has bicarb and mucus. The bicarb there gets secreted through these bicarb transporters, okay? And where do you get this bicarb from? From the blood itself. And where does the blood get the bicarb? Because it gets secreted at the same time as HCl. So as HCl, which is acid, gets secreted in the stomach lumen, bicarbonate, okay, will get secreted in the blood and it will go through the transporter, okay? So this is how the blood layer like protects, is a form of protection, if that makes sense, okay? And this is the neutralization, okay? This is what I meant by the neutralization of acidity, okay? You can also like refer back to slide 13. Let me go back to slide 13, okay? This is the mechanism of how bicarbonate gets secreted, and I believe, like, I'm not going to be covering it now because it was covered in other lectures, but if you have questions on it, text me later, okay? Or like email me. I'll add my phone number at the end. Okay. So is that clear so far? Is this how bicarbonate that's secreted into the stomach is formed, or is that something else? You mean bicarb that gets secreted? I believe that this is how bicarbonate gets secreted, yes, okay. And I'm sure I think there's another mechanism too from the cells themselves, okay. But again, this was, I think, covered in secretions lecture. I will go for it, like I will look into that and I will get back to you at the end, okay? Like, yeah, okay. You can like refer to your other lectures, and you can like text me and email me for like further questions, okay? Okay. So now, what happens if all three layers break down? Do you guys want a quick recap of the three layers of protection? Or is it clear? Recap? Okay. Let me just go back to the main slide. Okay, let's go back to the slide. Okay, so there's noxious material, bacteria, acidity, whatever it is that harms our stomach, okay, can be found in the stomach lumen, okay? But all of this, we don't want it to reach to our, like, we don't want it to reach like our stomach, okay, the actual stomach lining. Why? Because it can harm our body. And we will later on, as we get along through this lecture, we will explain how it can harm our body, okay? But it can cause harm, okay? So what does the stomach do in turn, which, like, how does the stomach defend itself from all of this damage, okay? It has layers of protection, okay? There are three layers of protection: the pre-epithelial layer, the epithelial layer, and the subepithelial layer. Khalid, I'll answer your question after I explain, okay? Okay. So, as I said, it protects itself using the three layers: the pre-epithelial layer, the epithelial layer, and the subepithelial layer, okay? The pre-epithelial layer contains mucus, which is viscous, and bicarbonate, which is the neutralizer. Remember when we said the mucus layer itself has two layers. They're functional layers. They're not anatomical layers. Okay, they're not like there isn't an actual line between them. Okay, but it's the inner dense layer, mucus layer, and the outer loose mucus layer. Okay, this is related to the pre-epithelial layer. The epithelial layer. Okay, this one, which is the second line of soldiers. There are two important points. Okay, the tight junctions, which prevents paracellular efflux. Okay, and cell repair mechanism. What's the cell repair mechanism? Which is restitution and complete healing. Okay, and then we have the subepithelial layer, which is through the last layer of protection, and this is through the blood flow. What cells secrete bicarbonate into the blood? Give me a second. I will search it because I don't want to give you the wrong information. Okay, I think that you took this in, what is it called? In the cell secretion lecture, right? Did you guys take this in the cell secretion lecture? Okay, okay, if I'm not mistaken, there's like acinar cells, okay, that secretes CL. Okay, Khalid, okay, can you email me, or during the break, I'll give you guys a small break because I don't want to pause this and answer the search now, okay, but I will note it down and I will get back to you eventually. Okay, let me write it down that I have to answer your question. Okay, I'll answer your question later. Okay. Okay, so what happens when all the 3 layers get broken down? Who can guess? Actually, I feel like you're guessing may be too long, but I'll just tell you. Okay, so after very noxious or very toxic, alters basically, but we'll get to that later. Okay, so after a very toxic stimulus, okay, breaks down all the 3 layers, okay, of the protective system. Yes, sepsis can eventually occur too. Okay, sepsis mainly like due to bacteria. Okay. After a very toxic stimulus breaks down all the layers of the protective system, which are the 3 layers that we discussed, okay, the acid eventually will reach the gastric mucosa, okay, as we covered in like as covered in like previous lectures, I believe. Okay, wait, let me just show you. Okay, so let's say first point. Okay, think about it in 3 points. Okay, first point, there's a breakdown of all layers, of all 3 layers. Okay, what will happen? Look at point number 2 now. Okay, so injury, breakdown of all layer. Well, what happens, I believe, is like, I think it was covered in previous lectures. Okay, acid will activate something called the CGRP. Okay, what's the CGRP? We have the calcitonin gene-related peptide. Okay, this is the calcitonin gene-related peptide. So acid manages to go through all 3 layers. Okay, acid, okay, itself will activate something called the CGRP, which is the calcitonin gene-related peptide. Okay, what does this? What happens after this CGRP gets activated? Okay, this will cause hyperemia. Okay, what's hyperemia? Hyperemia is basically increased amount of blood. Okay, and I will mention why in a bit. Okay, so it will cause hyperemia by nitric oxide. The doctor itself didn't emphasize the like specific mechanism like of nitric oxide, but best just know that all 3 layers are broken. Acid manages to get to the mucosa layer. Okay, the gastric mucosa. The acid itself that gets to that layer will activate something called the CGRP. Okay, which is the calcitonin gene-related peptide. The calcitonin gene-related peptide eventually will cause hyperemia, which is increased levels of blood. Okay, increased amount of blood. Sorry. How does it do that? Through nitric oxide. But to me, like, I don't think that's like very relevant to know through nitric oxide. But I believe like you know that nitric oxide causes like vasodilation, which is like dilation of the blood vessels to increase the amount of blood. Okay, that's how hyperemia occurs. Basically, when there is hyperemia, what will happen? Remember when we just mentioned that bicarbonate will be secreted into the blood as a 3rd layer of defense? You guys remember? Let's hope you remember. Okay, yeah. So for those that don't remember, this is the lecture. This is the slide that we were talking about. Okay, so when bicarbonate gets into the gets secreted into the blood, okay, alongside nitric oxide, okay, the amount of bicarbonate delivered, like the amount of bicarbonate delivered and mucus production will increase. Okay, so remember here, okay, when we said that bicarbonate gets released in the blood will carry it and help, and the bicarbonate will help in protection, because we know that bicarbonate is good in helping us protect, like protection against damaging forces, right? Yeah. Okay. So what happens when hyperemia occurs? There's more amount of bicarbonate allowed in the blood, right? So the more amount of bicarbonate in the blood, the more bicarbonate will be able to get secreted into the lumen, okay, through the bicarbonate transporter, okay, resulting into more protection. Okay, so when acid gets secreted into the lumen, bicarbonate gets secreted into the blood. We know this, right? Yeah, right. Okay. So in general, let's say the 3 layers get broken down, okay? The acid that reaches the gastric mucosa after the 3 layers gets broken down. By the way, the king is the gastric mucosa. We don't want anyone to reach the gastric mucosa because it can cause our body harm. So when all 3 layers break and the gastric mucosa, when the acid reaches the gastric mucosa, okay, the acid will activate something called the CGRP, okay, which is the calcitonin gene-related peptide, okay. Calcitonin gene-related peptide will cause hyperemia, more amount of blood. What happens when there's more blood? More bicarb, because we know that bicarbonate gets released in the blood. In turn, we will have more protection because bicarbonate gives us protection, okay? Yeah, so it will increase the amount of bicarb delivered and more mucus production, so more neutralization of acid, which equals more protection. The more mucus in bicarbonate, we have, okay, the more we create an ideal environment for restitution and healing too, okay? And this in general is how bicarbonate or the 3rd layer and everything helps us in protection, okay? Does that make sense? Okay. Perfect. Okay, let me move on now. So no one has any questions about this slide, right? We can move on. Nitric oxide stimulates mucus release. No, nitric oxide, I mean, like, yes, see if like in turn from here, it can stimulate mucus and bicarb release, but nitric oxide mainly has an effect on blood vessels in the sense of it causes vasodilation, which is like it dilates the blood vessel, okay, which causes hyperemia, which is like more amount of blood, okay. And when we have hyperemia, when we have like more blood, we will have more bicarb, and bicarb is used as like in protection. Does that make sense, Salma? What causes mucus release then? Nitric oxide does have a mucus release like mechanism, but mucus gets released from the own mucus cells. I think this was a question asked before, but the gastric mucosa, okay, it's composed, I think you guys took this before. Did you guys not take this in the gastric secretion lecture? I believe that you did. Okay, but just for like, because this isn't the focus of this lecture, but I think you'll be studying this in other lectures, but just for like quick recap, okay? The gastric mucosa, okay, is composed of many different types of cells, parietal cells. I feel like parietal cells is the only like main one I know, okay, but like it's composed of many different types of cells, okay? The ones that release acid and everything. There is a cell, okay, that's specifically to like for the release of mucus, okay? I think in the stomach it's the goblet cells, if I'm not mistaken, but don't like quote me on that. I will during the break, okay, I will search for what specific cell secretes mucus. But to answer basically your question, what causes mucus release, there's a cell specifically to release mucus, and also I'll answer Khalid's question, okay, about which cells secrete bicarbonate. Okay, one is the break in a couple of slides. I'll give you like a break eventually, okay? Okay. But other than that, are we good to move on? Okay. Okay, so we have one more defense force, which is the prostaglandins. So other than the 3 layers of protection, okay, we have one more defense force and prostaglandins, okay? I'll talk about what prostaglandins are now. So, just give me 5 seconds. I just need to quickly add something. I forgot to add it. Okay, I think I'll just explain it, okay? So, what are prostaglandins, first of all, before we include the rules? So does anyone know what prostaglandins are? You can type it in the chat. Yeah, perfect. Good job, Sihan. Okay. So what are prostaglandins? Okay, prostaglandins, they are a group of lipids, okay? And it's like, yeah, a group of lipids, okay, with hormone-like actions that are like secreted basically, or that your body makes, okay, in response to tissue damage and infection. Okay. So this slide, I feel like it was the perfect slide that I got from Dr. Peter, because it includes, like from his lecture itself, because it's just summarized by Dr. Peter, and I believe this is all he wants you to know on what like the function of prostaglandins. Okay, so prostaglandins are good. We want, like, in when we have infections, we want prostaglandins. Okay, why? Because they remain, or they maintain, the mucosal integrity. How? Okay, so other than the 3 layers of defense that we talked about, the pre-epithelial and subepithelial, we have something called prostaglandins. Okay, prostaglandins, they increase the release of mucus, okay, of mucus and bicarbonate. Okay, and we know that mucus and bicarbonate, they're good in the sense of they help us protect. Like, that's all we've been talking about so far, is the importance of mucus and bicarbonate. So they increase the release of mucus and bicarbonate. They also inhibit parietal cell secretion. What do we know about parietal cells? Parietal cells, they are the ones that secrete acid, okay? Or like, they secrete HCl, okay? And HCl is acid, basically, okay? And from this entire lecture, I think you got that we don't want the acid to reach the gastric mucosa because it can harm it, okay? So the acid is really good in the stomach, because we know, I asked you guys, you know the function, you said yes, it basically breaks down all the protein, whatever, okay? But we don't want it to reach the gastric mucosa, okay? So prostaglandins inhibit parietal cells from secreting HCl, okay? By gastric mucosa, do you mean the layers of the epithelial cells and the muscular and stuff? The gastric mucosa, I mean, it's, yeah, basically the answer to your question is yes, it's the lining of the stomach. That's what the gastric mucosa is, just the lining of the stomach. Does that make sense? Do I answer your question? Just the cells that line the stomach, that make up the stomach layer. Okay. Okay, so these are the, I mentioned the first function of prostaglandins and the second function of prostaglandins, okay? The third function of prostaglandins is that it maintains the mucosal blood flow. We just said we want mucosal blood flow, okay? Because the blood gives us more bicarb. Basically, like, the more the blood flows, the better, okay? And it also maintains epithelial restitution. Restitution is part of the like, healing, you can say, okay? So in general, everything so far that we mentioned in all layers, remember, like everything, the mucus, the bicarb, the mucosal blood flow, and the restitution, okay? And the fact that we don't want HCl to reach, or like acidity, okay, to reach the gastric mucosa, all of these factors that help us maintain the mucosal integrity are enhanced by prostaglandins. So when we have prostaglandins, like when prostaglandins get secreted, they will increase mucus and bicarb. They will decrease HCl by inhibiting parietal cells, and they will increase mucosal blood flow and epithelial restitution. This is everything that Dr. Peter wants you to know about prostaglandins. Is that clear? Are there any questions on the slide? They're released by which cell? You don't need to know. It's like completely, like, they have their own, like, it's not like, it's a different mechanism. You don't need to know. You just need to know that there's something called prostaglandins, okay? And they have these functions. You need to know what prostaglandins are, okay? There's something called prostaglandins, and you need to know how they maintain mucosal integrity. This is what you need to know. Are they secreted only during injury? Yeah, mainly prostaglandins are secreted during injury. Okay, is that clear? Everything clear so far? Okay, so up till now, we discussed the 3 layers of protection, the 3 lines of soldiers, you can say. pre-epithelium, epithelium, subepithelium. We discussed everything related to them. And we discussed prostaglandins, okay? These four things, okay, the three layers and prostaglandins, they maintain mucosal integrity, okay? What does that mean? They are the defensive forces against the damaging forces. Damaging forces is the acid, HCl, any noxious material, bacteria, okay? So this is the basic physiology of all of them. So far, everything clear? I'll hope so. Okay. So since we talked about that, now I'll be giving you guys a small break, okay? And then we'll come back and we'll discuss pathophysiology. So like what can go wrong if all of these things get broken or like nothing, these like don't work, okay? So how long do you guys want the break to be? Is like 5 minutes good? Okay, 15 minutes, okay, I'll give you like 10 minutes. Does that sound good? Or like 7 minutes? 7 minutes? Okay. You guys can like go drink water or something. I'm also gonna go drink water. How long the main part of the session? Inshallah, and I feel like what we did so far, that was the main bulk of everything. Okay. The other things are just like pathophysiology, like inshallah they'll go by faster, but like who knows, like I feel like it's not, the other parts isn't like necessarily hard, they're just like pathophysiology. So inshallah it will go like by fast. It's just like, you know, I don't know how long it will take because like, you know, sometimes you guys have questions and stuff. Yeah. So yeah, now you guys can like, if you guys want in the small break, you guys can unmute yourselves, talk. If there's any questions that you guys want to ask. Also, do you guys want my phone number for like quick access? Because here I like included my email at the beginning of the slide, okay? At the introduction slide. If you guys have any questions related to this lecture, okay, or anything like in general, like about university or anything, you guys can email me at this email, okay? I'll also like write my phone number on the chat where you guys can like text me if you guys have any questions, because I think like I added slides, okay, that like I just wanted you guys to like, when you look at this presentation, I want you guys to fully understand. Okay, so like I included slides for you guys to go through on your own time, okay, because a lot of the concepts, like I feel like for you to fully understand this lecture, you need to have a background knowledge. And that's why I included these like small slides, like from, you know, like for example, slide 13 about like this. Well, I'll show you. Okay, for example, this slide, okay? This concept, this was already covered in like previous lectures, okay? But I just wanted to include it for you guys to like recap, okay? So if you have like any questions about like these small slides that I included, okay? I also included a lot of slides from like external resources that I'll show you all of it later on, okay? And I also included like slides from Dr. Peter's book, okay? If you have any questions about this or about the lecture itself or about like anything in general, you can contact me. I will add my okay, I'll add my phone number in the chat. Okay. I think it was Khalid, and if I'm not mistaken, it was like either Siham or Selma. One of you, like Khalid, you asked me about the bicarbonate, and either Siham or Selma, you guys asked me. Selma, you asked me about which muscle, which cells secrete, I think, mucus, right? You guys can text me on here because I don't have your information. Okay, you can text me on here, and I will like answer your questions, or you can email me, or if you guys want to stay at the end of the session for like any like questions, like extra questions, I can search on the spot and get back to you, okay? Because I don't want to give you like wrong information. I want to make sure of like my information first. I just meant, is there a specific stimulus that causes release of mucus? Like a hormone or a neurotransmitter? Again, this is part of like your secretion lectures, okay, which is the one before this, I believe, gastric secretion. So I'm not like I'm sure there is, okay, but I don't know the specific one. So I will like That's why I said, like, if you want to come to, like, text me or whatever, I will search it for you and I will get back to you. Is that okay with you, Selma? Also, guys, you feel free to like unmute yourself now. This is the break. If you want, no pressure. How do you guys think the lecture so far? Like, are you understanding? Is there something that you guys want me to like change in the upcoming lectures and in the upcoming part? Okay, perfect. Of course. Okay, I'm glad it's helping. Yeah, so I think this is my first like pause session. So if you guys like have any like feedback or anything, you can always like come back to me, like, or like give it to me. Thank you. Thank you so much. Okay. Also, alongside Dr. Peter's slides, okay, I really encourage you to go through my presentation. Okay. Thank you so much. Okay, thank you. I really recommend you guys to go through my presentation, as I included so many like at the end you'll see, but I included slides from like Amboss and like external resources and links. Okay, and I also like transcribed everything I said in the notes section. Okay, so like you guys can watch the recording and go through the. What's the thing of the plushie on your bed? Those that's the one on the left is Kaslan and the one on the right is Coco. Okay. Anyhow, so yeah, I just recommend you guys to go through Dr. Peter's slides though, and go through his book. Tutu is on the floor. I have a stuffed animal. His name's Tutu. It's on the floor. Thanks. I like him on the floor. Okay, I don't think that's the point of the session. You guys can text me about that later. Yeah, he definitely take up half my bed. I'll give you guys 2 more minutes of break and I will go back. Okay. Did you guys take foveolar cells? Do you guys remember anything from your previous lectures? I'm sure that you guys did. You must have. I'm sure, yeah. Because okay. Are you guys ready to start again? Okay, so Khalid and Selma, you're both here, right? Because I'm ready to answer your question. I searched it and yeah. Just Khalid and Selma, please write yes, because I want this question, like the answers, to like be directed to you since you guys were the ones that asked. Okay, Selma, I don't know if Khalid's still here. Okay, so I'll just answer your question, okay? So Khalid and Salma, they asked me what cells secrete bicarb and what cells secrete mucus, okay? I strongly recommend you guys, okay, to go back to your gastric secretion lectures and like throughout your histology, because this wasn't the scope of the like this lecture that I'm teaching you right now, okay? But I did search it, okay? And if I'm not mistaken, okay, from my memory from GI, you have like the gastric glands, right? Okay. And within the like gastric mucosa, within the gastric mucosa, okay? And well, okay, yeah. Wait, let me just make sure. Yes, okay. So there's something called foveolar cells, okay? And these are found in the gastric glands, if I'm not mistaken. But like, please go back to your other lecture, okay? Because I don't want to give you like wrong information, but this is from what I managed to like collect so far, okay? Yeah, just make sure of the information I'm saying from your other lecture, okay? But these foveolar cells, I think you know them as gastric or like mucus neck cells, if I'm not mistaken, okay? And these are cells or surface mucous cells, okay? And they produce mucus and bicarb, if I'm not mistaken. Yeah. And they're found, these cells line the gastric mucosa, and mucus neck cells, they're found in the necks of the gastric pits, okay? And these are the cells that produce mucus and bicarb, okay? You also have goblet cells, okay? No, but the goblet cells are in the gastrointestinal tract. But I think the goblet cells also produce mucus and secrete it into the stomach, if I'm not mistaken. Yeah. Okay, does that answer your question, Selma, or I think also Siham asked it, and Khalid? Okay, acetylcholine histamine. I'm sure acetylcholine histamine increased the production, okay? But also go back to your other lecture because I'm not sure, to be honest, what other lectures or like what exactly Dr. Peter wants you to know, because there's so many different stimulants, okay? So go back to your gastric secretion lecture and all your previous lectures, and I'm sure this was explained in further detail there, okay? I just answered the scope of your question about like which cells secrete it. So when they release acid, that also causes. Oh, you mean like that's a stimulus, right, Selma? Yeah. So bicarb will go into the blood from the neck, and then to the lumen, from foveolar cells in the gastric pits. Yeah, foveolar cells are also called mucus neck cells, if I'm not mistaken. They're either same things or different things, but they both secrete bicarb and they will go into the blood and then to the lumen. Yes. Everyone clear on that? I can move on. Should I move on? Okay, because we'll go on to like kind of like new concepts now. So is everyone clear on everything so far? Okay, great. Okay, so now let's move on and discuss pathophysiology. Fun. Okay, so first, wait. Yeah, okay. Just want to make sure. Yes. Okay, first, we'll be talking about peptic ulcer disease. So, what's a peptic ulcer? A peptic ulcer is a sore on the lining of your stomach. So, you know, the gastric mucosa that we've been talking about this entire time, that we don't want acid to reach to it at all. Okay, so if there's any like acid that reaches to it eventually, I will like explain the mechanism now, but I just want to like explain what a peptic ulcer is. A peptic ulcer is a sore on the lining of your stomach. Okay, the ulcer doesn't only like occur in the stomach, it can also occur like in your intestine or your esophagus. Okay, but I just want to like show you what it is. Do you guys want me to show you a picture maybe for you to like understand? Let me like get a picture. Like for you to like imagine it. Did I include a picture in the presentation? I don't think I included a picture in the presentation, but let me like get you a picture from Google really fast. Which screen can you guys see? Can you guys see my slides or like my Google images? Oh, okay, let me share my screen with them. Okay, can you guys see now Google images? Okay, so do you see this image here? Okay, basically it's a sore, okay? Where it's like it's a breakage basically in the mucosal lining. Okay, it will cause like the sore. Okay, you know when you get like an ulcer in here inside, it's the same thing in the stomach. Okay, that this is the normal stomach, and this is an ulcer. You can also get duodenal ulcer, but this is like stomach ulcer. They said like you can get ulcers in your small intestine and also in like your esophagus up here. Okay, like this is a real life image, but I don't think you guys will like understand the picture or something, but like yeah, this one also looks like. Okay, so I just wanted to show you that. Let me go back to sharing my presentation. Okay, can you guys see my presentation now? You guys can see the presentation, right? Yeah, okay. Okay, so that's basically what a peptic ulcer is. Okay, now what's the mechanism of a peptic ulcer? When you have a lot of acid, you know, when we said there's a lot of acid and it can break through all the three defensive layers of the protective layers. Okay, when acid manages to break through all of these layers, okay, you will have excess acid. Okay, this will cause activation of pepsin. Okay, and eventually you will have an ulcer formation. This is the basic mechanism of how an ulcer gets created. Okay, what are the causes? You can have high acid in pepsin. Okay, because we know that pepsin eventually or like high acid will convert like pepsinogen to pepsin and all of that. Okay, all of this will cause an ulcer. Okay, you can ulcers can also be caused by irritation, like any noxious material. That's what we've been talking about the entire time. Okay, peptic ulcers can also be caused by poor blood supply. Why? I want someone to tell me why poor blood supply can cause ulcers from your understanding of the lecture that we took so far. Exactly, too little bicarbonate. Okay,
and because we cannot transmit bicarbonate.
Yes, perfect. That's the reason. Okay, and like obviously many other reasons, because like obviously blood supply is very important because it will like provide like nutrition whatever, but like the main concept of this lecture is like bicarb and whatever. So in this specific case, yes, it's bicarbonate along many other reasons, but the answer I was looking for is too little bicarbonate. So good job, guys. Poor mucus production and poor mucus secretion, as we just like this entire scope of the lecture, we know that mucus is very important, like an important defense mechanism. Okay, so If we have little mucus secretion, it can cause us a peptic ulcer. And the fifth reason is H. pylori, okay? For those that attended Dr. Peter's lectures, you might have like a background on H. pylori, but for those who don't know what H. pylori is, don't worry, I'll explain H. pylori in a separate slide on itself, okay? So I just want you to know so far that these are the causes of peptic ulcers, and this is the mechanism of peptic ulcers, okay? Okay, so continuation. What parts of the body can peptic ulcers affect? It can affect the esophagus. As you remember the picture that I showed you on Google Images, you can get like ulcers in the esophagus, in duodenum, which is like part of the small intestine, and in the stomach itself. Okay, you can in the esophagus. Okay, you can get peptic ulcers through, or like they're called like peptic ulcers, through something called GIRD. Okay, you guys took GIRD in histo, right?
We took it in physiology too.
Okay, yeah, and like physiology. Okay, basically that's when there's like acid reflux. Okay, and it like irritates the mucosal like layer of the esophagus, and the same mechanism of like peptic ulceration in the stomach will happen in the esophagus, the same way in the duodenum. Okay. Who knows? Okay, the most common places that peptic ulcers like affects. Does anyone know? Okay, I'll tell you. Okay, the most common places peptic ulcers affect are the duodenum and the esophagus. Why? Does anyone know why? I didn't include why in my transcription, but like I just thought of the question. Does anyone know why? Okay, I'm guessing that everyone knows why. Okay, so basically, because acid, okay, is normal in the stomach. Yes, they're not used to acidity. Perfect answer. Okay. Okay, because we like the normal thing in the acid, like in the stomach, is acid. Like acid is normally found in the stomach, and the pH, of the stomach is around 2. Okay, but in the esophagus and the duodenum, it's not usually acidic. Okay, since we have a lot of acid in the stomach, the stomach in result has more protection. Okay, all of the layers of protection, the three layers of protection that we talked about, are found in the stomach. Okay, why? Because to add more protection against the acid. In the esophagus and duodenum, they don't have this like added layer of protection. Okay, or they don't have like as much like intense layer of protection against acidity, because they are not used to as much acid as in the stomach. Okay, that's why the most common places ulcers affect, okay, are the duodenum and esophagus. That's just a fun fact. Okay, is that clear? Okay, I'll guess it's clear. Like I'll guess it's clear. Okay, thanks, Selma. Okay, yeah. So, what are the symptoms of peptic ulcer disease? Okay, you can have epigastric pain. That's the main like most common like symptoms that you'll have, like stomach pain. Okay, epigastric pain. What are the reliefs? Okay, you can give antacids. Obviously, antacids mechanisms, like antacids are anti-acids, so they are against acid. They will block the acid through, again, this wasn't covered in this lecture, but I'm sure you'll cover it later, through like proton pump inhibitors and all of that, because we know proton pump inhibitors. Do you guys take proton pump inhibitors? I'm sure you did. You must have. Did you guys take them? Yeah, basically that's like antacids, like an example of proton pump inhibitors. They will decrease the protons, and we know that like it will help, like proton pump is what increases, like it's like in the mechanism of like acid secretion. I think you took this in the secretion lecture. But basically, all you need to know is that antacids is a relief form of peptic ulcer disease. And milk. Okay. Why does milk? Does anyone know why milk is can be given in peptic ulcers? Oh yeah. Okay. Yeah. Sorry, I just didn't hear you have that. Okay. Not like that. Yeah, it's like okay, it's like alkaline. No, it's not. Yeah. Okay, it's not. I wouldn't say it's like a very alkaline. I wouldn't say it's very basic. Okay, but it is. It's not. It's not acidic. Milk is definitely not acidic. So yes, that's the reason, but also because like, yeah, it coats the stomach lining. Okay, so it's like kind of like a layer protection. Okay. Anywho, you don't need to know that fact. It's just like you just need to know that it relieves antacids in milk. Okay, like Dr. Peter didn't even like emphasize on why. Okay, so how can you diagnose a peptic ulcer through endoscopy? Simple. Okay, so so what are our defense mechanisms against peptic ulcers? Okay, we have the alkaline mucus. So remember the mucus that we just like we've been discussing this entire time. Okay, this acts as a defense against peptic ulcers. So everything we discussed so far is like the three mucosal like layers and the three layers of protection and like gastric integrity and all of that. This helps us against like it's a defensive like factor against peptic ulcers. Okay, so we have alkaline mucus, which helps us against gastric ulcers. Okay, so in the duodenum, okay, because all of that, by the way, all the three layers that we talked about so far, these are all in the stomach. Okay, and we know that like when we have HCl and pepsin, all this acid, okay, these are aggressive factors. Okay, these will these can like if they reach the gastric mucosa, they can cause peptic ulcers. And all these three layers in the stomach, they will protect against that. Okay, in the esophagus though, we have something called the submucosal glands. Okay, and there are very few of them, and they produce mucus. That's the protection in the esophagus. Okay, and in the duodenum, we have something called the mucosal or submucosal Brunner's glands. This is what you guys are talking about. Okay, so for those who asked me, what does how does mucus get secreted? This is how mucus gets secreted. Okay, through and we also have something called the goblet cells. I'm sure you guys are familiar with those. This is what the mucus can also get secreted through goblet cells and also bicarb, making that gradient. Okay, you know the pH gradient where bicarb adds like pH, like increases the pH. Sorry, against the acidity, which is low pH. Okay. The goblet cells here, though, are less developed than in the stomach. Okay, so there's more like this the action of like the goblet cells and like the gradient is mainly found in the stomach. Okay, but it's also found in duodenum, and we also have pancreatic release of bicarb, which is also like a defense factor. Okay, so that's the first defense like mechanism about like against peptic ulcers, other than all the other mechanisms we discussed. We also have sphincters. I'm sure you guys took everything about sphincters and anatomy and whatnot, okay? But sphincters, they act as, you guys know what sphincters are, right? Can I repeat the first part? The alkaline part, you mean? This one, the alkaline mucus. Is this the part you want me to repeat? Okay, so basically what this part is saying is that all of these cells, the submucosal like glands and the Brunner cells and the goblet cells and pancreatic bicarb, okay, all of these, they will release mucus, okay? And some of them, such as the goblet cells and the pancreatic cells, okay, they will release bicarb. And this will act as a defense mechanism against acidity. Okay, we'll add a layer, like an additional layer of protection. You know, everything we discussed before about mucus and bicarb, about how it like, it's protective against acidity. That's basically it. Is that clear? Did I answer your question? Okay, so since you guys know what sphincters are, you know that it's like they're basically like kind of like muscles that close and open, right? Okay, so the main point of this slide, okay, is to tell you that sphincters, they minimize, like upon the closure of sphincters, okay, they minimize the acid, like the esophagus and duodenal exposure to acidic chyme. Okay, so basically when sphincters close, acid in the stomach, like let's say this is the esophagus, okay, here's the stomach and here is the duodenum, okay, and we have sphincters here in between them. When sphincters close, all the acid will remain in the stomach and it will have a harder time going to the duodenum or the esophagus on the duodenum. So yeah, the esophagus and the duodenum won't be exposed to acid. So it will minimize the harm of acid, leading to, and this is basically a defense mechanism against ulcers. Okay, is that clear? Okay, so how does it happen? Okay, when the I feel like this is just like clear so far. Okay, here they're talking about pressure differences. Okay, so when there's a pressure difference, okay, that's how they will open. For example, let's say something's closed, there's so much pressure here, this will cause this to open. That's just basically what they're talking about, the pressure differences. Okay, and eventually the closure. It's just all of it is pressure differences. Okay, anyhow, so but the main point for you here to know is that sphincters can act as a defense against aggressive factors, mainly acid. Okay, if the lower esophageal sphincter, LES, okay, stands for lower esophageal sphincter. Okay, if the lower esophageal sphincter is defective, okay, let's say this sphincter doesn't work, what can this lead to?
Gastric, GER?
Yeah, GERD. Okay. Yeah, gastric esophageal reflux disease. Okay, that's what GERD stands for, I believe. Okay, and that's basically when the acid here, so this sphincter doesn't work, the acid in the stomach can go up to the esophagus and irritate the mucosal layer, okay, which is the lining epithelium of the esophagus, and can cause GERD. Okay. Okay, now we'll be talking about hiatal hernias. Okay, what are hiatal hernias? Okay, the definition of hiatal hernia is the protrusion of any abdominal structure or organ into the thorax through a lax diaphragmatic esophageal hiatus. Okay, so do you guys see the left part of the esophagus? Okay, let's see. This is okay. So this is the stomach. Okay, and this is the esophagus. Okay, the outer thing, do you see where it says diaphragm? It's the same thing on this picture. Okay, this is the diaphragm. Okay, if there is a lax or the, oh, by the way, I just wanted to show you that this here, do you see where the esophageal sphincter is? Okay, this is the esophageal sphincter. Okay, let's say, and wait, before I continue, I'm just gonna say this is the esophageal sphincter, and we have something called the diaphragmatic esophageal hiatus. Okay, which is the this space between the diaphragm and the esophagus. Okay, if this place is lax, means it's defective. Okay, it's not as strong anymore. It's loose. It doesn't do its function. Okay, its function. Okay, let's say this is the water bottle. This is the me holding it here. This is the I'm forming the diaphragmatic or the lower esophageal sphincter, and around it is the thus forming the diaphragmatic esophageal hiatus. Okay, let's say it's lax, it's so loose. Okay, what can happen? The material here, okay, since it's loose, I'm not holding it anymore. I'm not holding it in place. I'm not holding this part in place and this part in place. What happens if it's loose? The part in the lower and in this area can go up. Okay, so as you see, do you see how tight it is here? It's very tight like this. But do you see the distance now increase, making it more lax? Okay, meaning a part of the stomach, okay, can go up. Okay, do you see how this is part of the stomach? It can go up. Okay, because before it was only the esophagus on itself, and then we have this lower esophageal sphincter through the diaphragmatic esophageal hiatus, and then we have the stomach. But the stomach can be displaced, okay, here, if the diaphragmatic esophageal hiatus is lax. Okay, what's the risk factor? A major risk factor for this is obesity. This obviously this isn't the only risk factor, but the major one you have to know in this lecture is obesity. Okay, what can happen? I should have covered this part. Okay, but what can happen if part of the stomach, okay, goes up into the or gets displaced? GERD can happen. Okay, how can GERD happen? Because it's so lax, okay, to the point where it's not stopping the acidity from going up. You know how I said like here, it's usually like it's so like it's usually like not lax. It's usually like firm and keeps the acidity in place unless we want to open it, obviously. Okay, but if it's lax, okay, nothing. I'm not like nothing is basically stopping the acidity from going from like going from its place, which is the stomach, to going up. Okay, this basically the stomach got displaced. Okay, and obviously when this can cause GIT. Okay, and where's the location? The location of the esophagus. And this is the esophagus, and this is the stomach. Is the hiatal hernia clear? Is this also affected by lower esophageal sphincter? Aslan, the lower esophageal sphincter here will be displaced. Do you see the small dots here? The dot on the left and the dot on the right, or where the esophageal sphincter is basically like, it says where the esophageal I can't speak. Esophageal sphincter is. Okay, do you see the line? This is basically where the esophageal sphincter is. Okay, so Aslan, it will be displaced from then all the material is up. What causes this? Obesity is a major risk factor. This is all you need to know so far. That obesity can cause this. Okay, clear? Perfect. Okay, now moving on, we're going to talk about H. pylori. And for this part, I need your full attention. Okay, because it's not the fact that it's hard, but I think this is a new concept for many of you. Okay. Okay, so what is H. pylori? Okay, H. pylori is a bacteria. Okay, it stands for Helicobacter pylori. Okay, it's a bacteria that invades the stomach and is associated with peptic ulcer disease. Okay, H. pylori can cause peptic ulcer disease, but it's also like it's a bacteria. Okay, 50% of people are actually infected with like H. pylori, believe it or not. 50% of the entire population has H. pylori, but they remain asymptomatic. Okay, so I'll just give you the definition. I just want to quickly say, usually acid and pepsin in the stomach kill the bacteria. Okay, you know how like we have so many noxious material that goes into our body. Okay, and our body is made to protect itself. Okay, you know. So we have the stomach acid. You remember when we first said about like in the recap what the like part of the HCl is, or like the function of like the stomach is, okay, or acid in the stomach is, sorry. Acid and pepsin, they have bactericidal mechanism. Okay, they can kill the bacteria. Okay, but sometimes the bacteria can like invade. That's how like infections like happen, you know. But even if like as little as 1% of this bacteria survives, okay, it can replicate and cause an infection leading to peptic ulcer disease. Okay, so even as little as 1% can, this bacteria can like evade the system, and they can like replicate and cause peptic ulcer disease. Okay, but 50% of the people actually have, but they just are like infected, 50% of bacteria are 50% of people are infected, but 80 to 90% of these people that are infected are asymptomatic. Okay, so in reality, only like 10 to 20% of people like have the manifestation of like the disease. Okay, and 80, I don't want the statistics to confuse you. Okay, but 80 to 90% with people who have ulcers are like have H. pylori infection. Okay, so let's say like there are people like there's a person like people that have ulcers, okay, peptic ulcers, 80 to 90%, okay, of these people are most like are likely to have H. pylori infection. Okay, and yeah, okay, so as we just said, usually acid and pepsin kills the bacteria, but if even as little as 1%, okay, survives, it can replicate and cause an infection leading to peptic ulcer disease. Okay, how does this happen? This is, I need you to guys concentrate very much on this part. Okay, how can H. pylori cause peptic ulcer disease? By 3 mechanisms. Okay, 3 main mechanisms of H. pylori can cause peptic ulcer disease. Okay, the first mechanism is H. pylori has flagella. Okay, do you guys see this is an image of H. pylori? Do you see these like lines? Okay, this is flagella. Okay, this flagella gives H. pylori bacteria the motility. Okay, motility means like movement. Okay, and it lets it penetrate the mucus layer. You know how we said that H. pylori or like bacteria in the mucus layer find it very hard to penetrate because the mucus layer is very dense and thick, and yeah, and all because of also the ER and motility that gets removed, right? We mentioned this, right? Okay, but H. pylori has an advantage. It has flagella. Okay, so flagella can help it invade into the invade the mucus layer. Invade the mucus layer, and it can help it penetrate the mucus layer. That's the first mechanism of how H. pylori can cause peptic ulcer disease. Okay, the second mechanism, okay, it has spatial pH orientation. I don't mean to confuse it by this term, but I'm going to explain it now. Okay, just know that the first one is flagella. The second one is spatial pH orientation. Okay, what is spatial pH orientation? That it can detect where the acid, okay, believe it or not, but H. pylori, through this like spatial pH orientation, it can actually detect where the acid is and avoid it, okay? Because acid kills bacteria. And through like this spatial like pH orientation, it can detect where the acid is and avoid it, okay? Think of it like a ninja, okay? H. pylori is a ninja, okay? It has moves, you know how ninjas have moves, okay? It has moves through its flagella. Okay, so it's very sneaky and like it can move around, okay, and penetrate the mucus layer. And it's also very sneaky and secretive as it avoids the acid through spatial pH orientation. Okay, this is the first and the second mechanism. But we also have a third mechanism of like H. pylori and how it can cause peptic ulcer disease. Okay, the third mechanism is through urease, and this is what we'll be talking about now in this lecture, in this slide, sorry. Okay. Okay. So first of all, okay, if they avoid acid, how do they get killed? Okay, how does H. pylori get killed? If the person is infected through like with H. pylori, okay, and it gets to the point where it's severe and causes peptic ulcers, and all of the, you know, the layers of mucosa get like broken. You know how we said like invade the what is it called? Because where is acid? Acid in the stomach, right? Okay, so for H. pylori to actually cause the peptic ulcer, it needs to reach the king, so-called king. Do you remember? It needs to reach the like gastric mucosa, the epithelial lining of the stomach. Okay, it needs to evade the acid, which is like step number one. Okay, it needs to evade the first line of soldiers, okay, which is the pre-epithelial layer. It needs to evade the second line of soldiers, which is the epithelial layer, and the third line of soldiers, which is the subepithelial layer. And then once it reaches the stomach lining, okay, then it can cause a peptic ulcer. Okay, and why do only 50%? And that's why it's like with all of these steps, like we have so many protective like mechanisms in our body, which is like these three like layers, okay? All of this, okay, this will protect us from like H. pylori, this H. pylori bacteria and all like in many other types of bacteria, but in this lecture we'll be focusing on H. pylori, okay? Only then it can cause a peptic ulcer, okay? And that's why 50% of the population have H. pylori, have the H. pylori bacteria, but they're asymptomatic. Why? Because we have all of it, like everything we talked about so far about protection, that's still intact. Okay, they will provide protection against H. pylori bacteria, but only then all of like everything's breached, okay, and it reaches the gastric or the stomach lining, the gastric mucosa, okay, then it can cause a peptic ulcer, which is the sore that we're talking about, the definition of what peptic ulcer is, okay? With all of that being said, okay, you can, the body will still have like other defense mechanisms, okay? If then and only then it invades like everything, and the body cannot do anything to like protect itself from the H. pylori bacteria, then obviously this person will have like severe peptic ulcers, okay? And they will feel like severe epigastric and like stomach pain. They will go to the hospital and then doctors will like treat them with medication. Is that clear? Did I answer your question? Okay. So we said H. pylori can cause peptic ulcers through 3 mechanisms: flagella, pH spatial orientation, and the third one through ureas. Okay, now I'm going to be talking about we discussed the first 2. Now we're going to be talking about the third one, which is ureas. Okay, first let's explain what urea is. Okay, urea is everywhere in the body. Okay, it's a very small molecule. Okay, that's made up of nitrogen, carbon, and hydrogen. Okay, I want you to remember the components of urea because they're important for you to understand the mechanism of how H. pylori can cause peptic ulcers. Okay. So urea is a small molecule that's made up of nitrogen, carbon, and hydrogen. Let me write them in the chat so you guys don't forget. But everything is written in the what is it called? In the notes section. I transcribed it for you guys. Okay, so urea is a small molecule that's made up of nitrogen, carbon, and hydrogen. Okay. What breaks down urea? We have something called urease. Okay, urease is an enzyme that breaks down nitrogen, urea, sorry. Okay. So what does H. pylori happen to have? Urease, okay? H. pylori happens to have urease, okay? So what happens is that when urea, okay, which is everywhere in the body, okay, enters the H. pylori molecule. Do you see this? The pink dot here? This is urea, okay? Let's say it enters into the H. pylori molecule, okay? What is it called? H. pylori, okay, it has a gate for urea. Okay, because H. pylori likes urea. Why? Okay, because it, what is it called? It has a gate, and I'll explain obviously why now, okay? When H. pylori gets into the when urea, sorry, gets into the H. pylori bacteria, okay, it gets broken down into ammonia, okay, you know, we said that urea is made up of nitrogen, carbon, and hydrogen, okay, but when urea gets into the bacteria through urease, okay, urease is what breaks down urea. When urea gets into this H. pylori, urease, which is already found in H. pylori, will get broken down into ammonia and carbon dioxide. Okay, is that clear so far? Do you see? Urea gets into the H. pylori through the urea gates, okay, and urease, which is already found in H. pylori, will break down urea into carbon dioxide and ammonia. Okay, why did I say it was important for you to understand what makes up urea, nitrogen, carbon, and hydrogen? Because nitrogen and hydrogen is what makes ammonia, and carbon obviously is what makes carbon dioxide. Okay. Okay, so why does urease have? Why does H. pylori have urease? Okay, we just said that urea is everywhere in the body, and in H. pylori, okay, it's very sneaky. Okay, it wants to use everything around it, okay, to basically survive. Okay, so H. pylori uses urea, okay, to eventually cause peptic ulcers, okay, through urease. Urease is already found in H. pylori, okay? The bacteria itself has urease, and it will break down urea into ammonia and a carbon dioxide. Now you might be thinking like, okay, and like how does ammonia and carbon dioxide help H. pylori flourish? This is what we're going to be talking about now. Okay, so concentrate. So ammonia happens to be basic. Okay, ammonia, which is basic, leaves the bacteria. Do you see? This is ammonia through these like gates of like the pink gates. Okay, ammonia leaves the bacteria and forms something called the ammonia cloud. Okay. All of this, do you see the triangle, the purple triangle? This is ammonia, okay? The purple triangle, ammonia, leaves the bacteria and goes all around the bacteria, okay? Forms something called the ammonia cloud, okay? What's the ammonia cloud? It's a buildup of ammonia around the H. pylori bacteria, okay? What does this do? Ammonia is basic, okay? So it increases the pH around the bacteria, okay? It's so sneaky, okay? Increases the pH around the bacteria, making it evade the acidic environment, okay? Which we know is bactericidal, okay? So acid is bactericidal, okay? H. pylori, okay, doesn't want to get killed. It wants to like spread around and cause an infection, okay? And causing a peptic ulcer, okay? How does it do that? It transforms, okay, or like breaks down urea into ammonia and carbon dioxide by urease, okay? This ammonia leaves the H. pylori and surrounds the H. pylori bacteria, okay, forming an ammonia cloud. And ammonia is basic, okay? So the fact that it's basic, okay, around the H. pylori, it's not, the acid isn't going to reach the bacteria, okay? So that's how it can evade the acidic environment, okay? Creating the optimum environment for the H. pylori bacteria to flourish, okay? The ammonia cloud not only forms the basic environment around the bacteria, helping it like evade the acidic environment so it's not going to get killed, so the bacteria is going to flourish, but it also helps the flagella, this part of the bacteria, the flagella, move as the acidic environment inhibits the flagella. So the acidic environment, okay, kills the bacteria and inhibits the flagella. When urea through urease gets converted into ammonia and carbon dioxide, the ammonia forms the ammonia cloud around the bacteria, okay, helping it evade the bactericidal environment, which is the acidic environment, okay? So the bacteria isn't going to get killed, and the flagella isn't going to be inhibited by the acidic environment. Is that clear? So far. They don't have questions about H. pylori. Perfect. Okay, guys, this concept is very important, as well as every other concept, but like this H. pylori, you'll definitely get questions on that in the exam, okay? As well as the 3 gastric mucosal defenses, you'll definitely get questions on that in the exam, okay? But I remember like there was like H. pylori is very emphasized, like he emphasized on this a lot, okay? As well as the other like pathophysiology things that we'll be discussing later on this lecture, okay? So let's say a patient has H. pylori and H. pylori manages to cause a peptic ulcer, and it like causes an infection and evades all the like protective mechanisms, okay? How do we test for H. pylori? Like how do we make sure that this peptic ulcer in the stomach of the person is caused by H. pylori? Because as you remember before, we said that, where is it? Okay, peptic ulcer has many causes, okay? But how do we know that H. pylori specifically is the cause of what causes H. pylori? Okay, we know through laboratory testing, through something called urea breath test, okay? So urea is important in helping us to detect H. pylori. How? By carbon 13 and 14 urea breath tests. We just said that urea is made up of nitrogen, carbon, and hydrogen, right? Keep that in mind, okay? So we can detect whether a person has H. pylori through the urea breath test, okay? What does this urea breath test do? It measures carbon dioxide to determine whether a person is infected with H. pylori or not, okay? Simple, just know the definition. Now we will explain the urea breath test. Okay, this concept is also important. So listen, okay? How does this test work? Okay, the urea breath test, is H. pylori the last concept? No, H. pylori isn't the last concept, but we don't have many concepts after that. Like this is the main bulk of the pathophysiology. After that, like it's the concepts aren't as like broad, like aren't as in detail like as H. pylori. Like H. pylori is the most detailed one. Everything like else is gonna go by. It's gonna go by like really fast. Inshallah. Okay, so how does this test work? Okay, the urea breath test is a non-invasive method where a patient is given urea with labeled carbon 13. Okay, we know, because go back all the way to chemistry, we know that there's something called isotopes. Okay, carbon-13 is an isotope of carbon, okay? Because originally carbon has an atomic, like, what is it called? The atomic number? Atomic mass, sorry, of 12, okay? But we give the patient, okay, urea with labeled carbon-13, okay, which is an isotope, okay? So urea is taken orally, okay, with labeled, like the people like in that are like in the clinic, okay, the carbon-13 is labeled, okay? The urea is labeled with carbon-13, like they make it in a lab or whatever, wherever they make, okay? The patient is given urea orally with labeled carbon-13, okay? Urea is taken into the stomach of the patient and it goes into the bacteria, okay? Remember here when we said that the urea goes into the bacteria, okay? This urea goes in, that's taken orally, it goes to the bacteria, okay? And if H. pylori is present, okay, if the patient has H. pylori, H. pylori will take this urea, okay, and it will use the urease in the H. pylori itself to break down urea into ammonia and carbon dioxide, as we explained previously, okay? This carbon dioxide, though, okay, it will be labeled with carbon with C13, okay, with the isotope that we labeled it previously because it was given orally, okay, because it came from the orally taken urea, okay. These will be taken, okay, the carbon dioxide and ammonia, okay, they will be taken to the blood, okay. Carbon dioxide will be taken to the lungs because it's exhaled, okay, while ammonia will be taken to the urine, okay. So we said that carbon dioxide is taken to the lungs because we exhale carbon dioxide, okay. When we breathe out carbon dioxide, okay, we take this carbon dioxide that this patient breathes out and we look at it, okay. If we see that the carbon dioxide that the patient like breathes out, okay, has the isotope of carbon-13, which is the one that we labeled, the carbon-13 labeled isotope, we know that this person is positive for H. pylori, as it came from the labeled carbon in the urea that we gave the patient orally, okay? Because carbon is usually has an atomic mass of 12. But if we see the carbon with the atomic mass of 13, we know that this is the labeled one, because in the body, usually carbon has an atomic mass of 12. But we use the isotope and we gave them orally. We gave the urea orally with labeled carbon-13. So if the patient breathed out carbon-13, we know that this patient has H. pylori infection. Is this clear? Is the urea blood test clear? Okay, perfect. This is also a very important concept. Okay, so it's not clear. What did you not understand? Like which specific aspect of the whole thing? Okay, how about you stay at the end and I will explain it to you in like detail, like step by step, because like I already explained it now and I don't want to like have to re-explain it now because it's going to take like a lot of time again. So stay at the end and I'll explain it to you, okay? Because we need to like move on, okay? Okay, so okay, H. pylori usually affects yeah, Sam, okay, I'm gonna explain it then. Okay, H. pylori usually affects the antrum region of the stomach. Okay, you guys took anatomy, you guys should have taken the like parts of the stomach, and just know that H. pylori usually affects the antrum region. Okay, but as it multiplies and becomes chronic means like you'll have the infection for a long time. Okay, it can cover the entire stomach. Okay, which causes a decrease in acid production. Okay, because H. pylori around it has so much like ammonia and whatever. Okay, we will like around the stomach, inside the stomach. Okay, if you a complete like the entire stomach will have a complete H. pylori like invasion. Okay, this will decrease this the acid in the stomach. Okay, remember that when we previously mentioned that a lot of people have the H. pylori bacteria but are asymptomatic. Well, this leads people to believe that it somewhat has a benefit. Okay, that H. pylori somewhat has a benefit, and its eradication will lead to gastric acid, will lead to gastric diseases such as like GERD, like Barrett's esophagus, and like esophageal cancer due to acidity. Okay, so what's important in this slide is the chronic infection, which is basically that chronic infection of H. pylori affects the entire stomach, not just the antrum. Okay, that's basically what the chronic infection is. And the eradication part is that a lot of people believe that if you get rid of H. pylori, okay, that since that H. pylori decreases the stomach acid, that if we completely get rid of H. pylori, okay, even in like the asymptomatic people, okay, that it will cause GERD, Barrett's esophagus, and esophageal cancer. Like H. pylori has been in like people's like stomachs for such a long time, like centuries back, okay? But in 50% of people, it's asymptomatic. Okay, so a lot of people believe that if in these asymptomatic people, if we get rid of H. pylori, it's going to cause us GERD, Barrett's esophagus, and esophageal cancer due to the acidity. Okay, that's the main concept of the slide. Okay, and this is a peptic ulcer disease summary from Ambos. I got it for you guys. It's very helpful, and I strongly recommend you guys read it. Okay, if you have any question on it later, you can text me or email me. Okay, but I'm not going to read it now. You guys read that on your own time. Okay, now I'm going to discuss NSAIDs. Yay. Okay, NSAIDs are non-steroidal anti-inflammatory drugs. Okay, such as aspirin and ibuprofen. I'm sure you guys know what aspirin and ibuprofen are, right? Okay, I'm sure. Okay, there are many drugs that are under NSAIDs, but an example of NSAIDs or non-steroidal anti-inflammatory drugs are aspirin and ibuprofen. Okay, the image here. Okay, let me first read like the mechanism of action. Okay, NSAIDs is like a topic on its own. Okay, you ask why do 80% of people that have ulcers have H. pylori infection then? Okay, that's why I didn't want the percentages to confuse you. Okay, 50% of the population has H. pylori, but are asymptomatic. Okay, not okay. Let me re-explain. So 50% of the people have H. pylori. Okay, and this 50% of people, 80 to 90% of these 50%, okay, are asymptomatic. Okay, that's one statistic. Let's go to the people who have.
Can you go back to the last slide?
The slide itself? The slide about the percentages? Which slide? The last slide? Wait. Okay. You mean this slide about the Ambas one? Okay, I'm guessing that you mean this slide.
Wait, I'm sorry, you were glitching.
Can you guys hear me?
Yeah, you're fine for me.
Okay. Okay. So, Khaled, to answer your question, okay, that...
Sorry, I was glitching, the net was bad. I was talking about the one with H. pylori and...
Okay. Do you have a question on that slide, the H. pylori slide? Which one? This one?
Yeah, the eradication. This slide, exactly. So when we eradicate the H. pylori, it's going to increase the GERD and the Barrett's esophagus, or what?
That's what people believe. Like, it's not like proven, yani, because we know that H. pylori is a bacteria and it's not good. But I only included it because the doctor went over it briefly. But yani, that's just a belief that people have, that if we eradicate H. pylori, that can cause any GERD. Like if we completely eradicate it, even the asymptomatic people, it can cause GERD, Barrett's esophagus, and esophageal cancer. To be honest, this concept is very low yield, not that important. You don't need to focus on this slide too much. Okay.
Okay.
Okay. Khalid, answering your question, why does 80% of people have ulcers have H. pylori? Okay. So we said that 50% of the population have H. pylori. Okay. 80 to 90%, okay, of these 50% that have H. pylori, 80 to 90% are asymptomatic. Okay, that's one thing. Okay, that's one statistic. Put that aside. Now, let's say people have peptic ulcers. You know how we said that peptic ulcers have so many causes? Where's the slide? You know how we said that peptic ulcers can be caused by like so many causes, okay? 80 to 90% of people that have peptic ulcers are due to H. pylori. Okay, I mean, not due to high acidic like high acid, not due to irritation. 80 to 90%. Like there's so many, all of these are causes, but 80-90% of people with peptic ulcers is due to H. pylori. Is that clear, Khalid? Is there a specific reason? It's just that H. pylori, like there isn't like not to my knowledge, there's a reason why, but this is just the most common cause of peptic ulcer disease. Like the most common cause, and well, I don't know why it's the most common cause, but just know that the most common cause of peptic ulcer disease, 80-90% is H. pylori infection, because it's like a bacterial infection, because like, yeah, it's already found in 50%, it's already found in the stomach, like in 50% of the people it's already found, okay? So as I remember, I said like even as 1% like can replicate, it can like replicate completely. So it's like a common cause, okay? Because let's say we have high acid and peptic. I'm gonna explain it from my understanding. Yeah. Okay. Let's say we have high acid and pepsin. Okay. The body has so much other mechanisms such as the blood supply and like mucus secretion that can help against high acid. Okay, and same for blood supply and same for mucus secretion. So eradication of H. pylori damages the esophagus, and having H. pylori might damage the stomach. Okay, this slide. Where is it? Okay, don't focus so much on the slide. Okay, this is just what doctor mentioned, and I thought was like I was gonna, because I don't want to miss any concept from the doctor. Okay, so like I just wanted to include it. Okay, it's what people believe. Okay, you know how we said that a lot of people with that have H. pylori, 90-80% are asymptomatic. Okay, people believe that if we remove the H. pylori from the people that are asymptomatic, okay, they can have GERD, Barrett's esophagus, and esophageal cancer. Okay, so this is the fact about this is what's like about the eradication. Okay, and having H. pylori might damage the stomach. Yes. Okay, it's so common for H. pylori to damage the stomach because 90-80% of peptic ulcers are due to H. pylori. Is that clear, Khalid? Yani what's really important about this is just the concept of how H. pylori can cause peptic ulcers through the three mechanisms we discussed, and especially the urea one is very, very important. The one that we discussed on this slide. This one is very important, okay? And urea breath test is very important too. It's very high yield, okay? As per this slide, this slide, if you literally want to skip it, in my opinion, to be honest, it's not going to really affect that much, okay? And this is a summary of peptic ulcer disease, okay? Read it, it's very helpful. Okay, so we now moved on to NSAIDs, okay? So NSAIDs are non-steroidal anti-inflammatory drugs. Examples of NSAIDs are aspirin, ibuprofen. Okay, what's the mechanism of action of NSAIDs? You don't need to know the mechanism of action in detail. I just want you to know that we have something called the cyclooxygenase enzyme. Okay, NSAIDs inhibit this enzyme. Okay, which leads to the inhibition of prostaglandins. Okay, so what Dr. Peter emphasized on this lecture is that the mechanism of action of non-steroidal anti-inflammatory drugs, which are NSAIDs, is the inhibition of prostaglandins. That's why I put it in italics. All you need to know is that it inhibits prostaglandins through the inhibition of the COX-1 enzyme. Okay? Okay, so this image, okay, or let me explain it in a different way. Okay, so what will we have? You remember the function of prostaglandins that we mentioned, the three main functions of prostaglandins, okay? In this slide, let me show you. Let me go back and show you the slide. This slide. Remember this slide? Okay, these are the functions of prostaglandins, the role of prostaglandins in maintaining mucosal integrity. Okay? Now, okay, if we have NSAIDs, okay, that inhibit prostaglandins, what will happen? All the function of prostaglandins, خلاص, they will be stopped, okay? So the effects will be decreased in mucus, decreased in bicarbonate, increased in HCl, decreased in mucosal blood flow, and decreased restitution. Okay. I want you guys to focus on this image here. You guys can all see the image, right? I'm guessing yes, because I can see the image. Okay. So this image shows the functions of prostaglandins and the effect of NSAIDs. Okay, remember the last slide I just showed you all the roles of prostaglandins. Okay, all of these roles will be inhibited by NSAIDs because NSAIDs lead to inhibition of prostaglandins. Okay, so do you see the red? This is the effect of NSAIDs. So we know that prostaglandins, we can see the slide. Okay, perfect. You know that prostaglandins increase the release of mucosal, mucus, and bicarb, right? We took this already before in the functions of NSAID. Now, if we gave someone non-steroidals, anti-inflammatory drugs such as aspirin or ibuprofen, okay, it will lead to a decrease in prostaglandins. What will happen? We will have a decrease in mucus and bicarb, since the function of prostaglandins is to increase mucus production and bicarb. If we inhibit this function, we will have a decrease in mucus and bicarb. The same thing with HCl, okay? NSAID's function, or what is it called? Prostaglandins function is to inhibit parietal cell secretion, okay? If we inhibit prostaglandins, it will increase HCl, okay? Because it will not inhibit the parietal cells anymore, okay? The same thing for blood flow, okay? If we decrease prostaglandins, mucosal blood flow and restitution will decrease, okay? So, as we previously mentioned, I'm going to go over this really fast again. As we previously mentioned, functions of prostaglandins are increase mucus and bicarb, decrease acid, okay, by inhibiting parietal cells, maintaining mucosal blood flow and epithelial restitution, okay? So if we give something that inhibits the function of prostaglandins, none of these functions will occur. This is exactly what NSAIDs do. Clear? Okay. So, but now you might be asking yourself, okay, if NSAIDs cause all of these damage, why do we still give them to patients? Okay, well, NSAIDs are really good pain relievers. Okay, we give them, but when we give them to patients, okay, we give them in moderation and with care to avoid the damage that may induce. Okay, that they may induce. Okay, this is why people who take NSAIDs are a lot more prone to getting gastric ulcers. Okay, clear? Okay, yes. Okay, so now I'll be talking about Zollinger-Ellison syndrome, okay? This should go by into a lot fast, okay? What's Zollinger-Ellison syndrome, okay? Okay, Zollinger-Ellison syndrome, okay, it's basically a tumor, okay, that secretes gastrin uncontrollably, okay? Or yeah, that can secrete gastrin uncontrollably. But okay, wait. Where is gastrin originally secreted from? If you guys go back to your gastric secretion lecture. Do you guys know? G cells, perfect, okay? Gastrin is usually secreted by G cells, okay? But in Zollinger-Ellison syndrome, okay, we have a tumor that secretes gastrin uncontrollably. It's not the G cells that secrete the gastrin in this case, it's the tumor itself, okay? What's gastrin? Gastrin is a peptide hormone primarily responsible for enhancing gastric mucosal growth, gastric motility, and secretion of hydrochloric acid, which is HCl, into the stomach, okay? So in Zollinger-Ellison syndrome, we have increased gastrin, but the gastrin here isn't from the G cells, it's from a gastrinoma, okay, which is a tumor. Gastrinoma is a tumor found in the pancreas mainly. So Zollinger-Ellison syndrome is mainly a like a syndrome of the pancreas. Here I included a picture of the pancreas so you guys like can remember, okay? And it's mainly in the pancreas, but it can only be it can also be found in duodenum, okay? This gastrinoma or the tumor, okay, it secretes gastrin, hence the name gastrinoma, okay? What are the effects? Okay, it can, I'm sorry about the sound, okay, I hope you can still hear me, okay? It can cause duodenal ulcers due to increase in duodenal acidity. Is the sound really loud behind me or no?
No, you're good.
Okay, alhamdulillah, okay? So the effect is duodenal ulcers, okay? Due to the increase in duodenal activity, because we said that gastrin can increase HCl. Okay, so the mechanism of action is increase in acid, okay, leads to increase in pepsin activity, okay, which leads to mucosal damage, okay? You know how we said that a gastrin can increase HCl, okay? When we increase HCl, pepsin activity will increase, which leads to mucosal damage, leading to peptic ulcer, okay? Eventually, okay? We can also have this, that's the first mechanism of action. The second way, we can have a decreased pancreatic enzyme activity because Zollinger-Ellison syndrome is mainly a tumor in the pancreas. So we will have, in results, we will have decreased pancreatic enzyme activity, which decreases lipase activity, okay, which causes there to be more lipids in this stool, and it will cause steatorrhea. Okay, this is just like something that will happen with like Zollinger-Ellison syndrome. Okay, so is Zollinger-Ellison syndrome clear? Like, what is Zollinger-Ellison syndrome? It's basically a gastrinoma, okay, mainly found in the pancreas that will release gastrin, and through all the functions of gastrin, okay, it can lead to duodenal ulcers due to increased duodenal acidity. Okay, clear? Okay, what's the treatment? Just to remove the tumor. Okay, it's not obviously, it's just as simple as like removing tumor, but like, obviously because it's like a disease, okay, but the treatment is to remove the tumor, okay? How did they discover this gastrinoma? Okay, and how do they know that this gastrinoma to the tumor? Okay, well, if you recall from previous lectures, okay, that there's a feedback inhibition through secretin, okay? When there's a lot of acid production, okay, there's a secretin will have like feedback inhibition, okay, that will stop gastrin, that will stop G cells from releasing gastrin, okay? So what did they do? They found these people with like increased like gastrin levels and and Zollinger-Ellison syndrome, okay? They found them to have like increased gastrin levels, okay? So they tried to inject secretin, hoping gastrin levels will decrease, okay? But they did not, okay? Gastrin levels did not decrease, okay, in these specific people when secretin was injected. So this shows that the gastrin production here isn't from the G cells themselves, okay, but from the tumor instead, from this gastrinoma instead, okay? Is that clear? Okay, right, yes, in the chat, okay, it's clear. Okay, perfect. Okay, here I included a summary from AMBOSS about Zollinger-Ellison syndrome, and I really advise you guys to read it, okay, on your own time. Okay, so now we got to our last concept, okay, which is vomiting. Okay, vomiting is also called emesis, okay? So in the notes, I included a video that I really recommend you guys to watch on vomiting, okay, physiology of vomiting. So vomiting, believe it or not, like as bad as it sounds, it's a protective mechanism. Like if you vomit, like if your body is the one that stimulates the vomiting, obviously if it's not like self-induced or anything, vomiting can be a protective mechanism that removes the irritants found in your GI tract, okay, in your stomach, like specifically, okay. So what's the physiology of vomiting? Okay. EC cells, which are enterochromaffin cells, okay, they release 5-HT, which is serotonin. Okay, so enterochromatin cells release serotonin in response to cytotoxic agent. Okay, let's say الحين I ate something bad. Okay, my enterochromaffin cells, okay, in my body, okay, they sense this, or like in my stomach, okay, they sense the cytotoxic agent. Okay, what will they do? They will release serotonin. Okay. This serotonin will stimulate the serotonin receptors on the sensory nerves in the vagus, on the vagus nerve. Okay, in the vagus nerve, this serotonin will stimulate the serotonin receptors. Okay, this stimulus will, when you stimulate this sensory nerve, okay, the stimulus will travel through the nerve fiber to the vomiting center in the brain. Okay, remember that it's to the vomiting center of the brain. Okay, and this will trigger the vomiting reflex, okay, which is the vomiting itself. Okay, so this is basically the physiology of vomiting, and I really advise you guys to watch the video I linked in the notes. Okay, so now what's the vomiting reflex? Okay, first we will have relaxation of the lower esophageal sphincter. And let's say all of this happens, like all of the physiology happens, and like my vomiting center in my brain triggers my body to vomit. Okay, how will this vomiting occur? My lower esophageal sphincter will relax. Okay, so relaxation of the lower esophageal sphincter. LES is lower esophageal sphincter. Okay, contraction of diaphragm and abdominal muscles. Okay, so when my diaphragm and my intra-abdominal muscles will contract. Okay, this will cause a rise in pressure, in intra-abdominal pressure. Okay, when the intra-abdominal pressure rises, okay, it will like push, okay, all the food upwards. Okay, because the pressure inside will be more than the pressure outside, and we know that from like passive to active, whatever pressure, it will go from high pressure to low pressure, right? So it will increase the pressure, okay, causing my epiglottis to close, because obviously we don't want food or like vomiting material to go back to the our breathing, okay, and stop like our breathing pathway. Okay, it will close the epiglottis, and all the food, because of this high pressure caused by the contraction of diaphragm and abdominal muscles, all of this high pressure will cause vomiting, which is the expulsion of food. Okay, clear? And I don't even think your doctor, Dr. Peter emphasized this concept a lot, but I think it's important. Yeah. Okay, so we don't only have the not the only okay, you know how we said that what triggers the vomiting is the vomiting vomiting center in the brain. Okay, the vomiting center in the brain isn't the only thing that triggers though. Do you see here? This is the vomiting center in the brain. Okay, but it's not the only thing that triggers vomiting. We can have something called the chemoreceptor trigger zone. Okay, it can also trigger vomiting. Okay, here we said what triggers the vomiting center is through this process. Okay, through the serotonin and the vagus nerve and everything. Okay, the chemo trigger zone. What are the triggers that trigger vomiting through the chemoreceptor trigger zone? We will have motion sickness. Okay, because a lot of people that when they get motion sickness, they vomit. Okay, and it's through the receptor trigger zone. Okay, drugs such as opiates and chemotherapy can also trigger the receptor trigger zone to induce vomiting. Okay, hormones such as in pregnancy, upper GIT irritants such as alcohol, and pain and emotional responses, like when people get anxiety or like when they feel like severe pain, it can also trigger vomiting, and this is all through the chemoreceptor trigger zone. Okay, I just want you to know that the basic concept here is that there are two ways, either through the vomiting center, which is this what we explained here, okay, and the chemoreceptor trigger zone. Okay, is this concept clear? Okay, right, and like yes in the chat if you find this concept clear. Okay, perfect. Now, if there are no questions on this, we will move on to our pop quiz. Okay. So I want you all to focus. There are a couple of questions, and I got some of these questions from what is it called? From what's his name? The Guyton test bank. Okay, the Guyton book that I think Dr. Peter recommends the Guyton and obviously his book. Okay, but the Guyton has questions, so I got a lot of questions from Guyton, and I also got them from past papers. Okay, so listen, because this is how you'll use all the material that I just explained in this past 2 hours and a half. You'll use it to answer questions. Okay, so let's move on to the first question. Okay, I want you guys to write your answers in the chat. I'll read the question, and I want you guys to write your answers in the chat, and then I'll explain each question. Okay. First question is, a 67-year-old man has heartburn and stomach pain. Okay. Endoscopy shows several small gastric ulcers in the corpus. Biopsies were positive for H. pylori, which promotes gastric ulcer formation by damaging which of the following? Okay, perfect. The answer is B. I think this is a very straightforward question, okay, because we know that anything that damages the gastro-mucosal barrier, which is everything we've been talking about this lecture so far, okay, the 3 layers, if everything gets damaged, ulcers formation can, like peptic ulcers can occur, okay? And we know that H. pylori is one of the main causes for peptic ulcers, okay? So this is how you'll answer the question. Is that clear? Is this question clear? Or do you guys have any questions about like other answers, or do I move on? Does anyone have a question about this question? Okay, so I'll assume that's clear and I'll move on to the second question. Okay, the second question, this is, okay, guys, in the notes section, by the way, I wrote, for example, the first question, I wrote in each question my source and what chapter and which question, so you can refer back to the question. Okay, so this happens to be from the Guyton textbook, chapter 64, question 2. Okay, I'm not going to mention the source for each one. You guys can look at it under my slides in the notes section. Okay, so the second question is, a 44-year-old man underwent a cholecystectomy, okay, 9 years ago. At that time, the liver was normal, but an enlarged pancreatic head was identified. 3 years ago, he was admitted for upper gastrointestinal bleeding. Endoscopy revealed multiple ulcers in the duodenum. The serum gastrin level was 1,100. Okay, the normal is 0 to 100, so it's severely the serum gastrin is severely elevated. Okay, abdominal computed tomography revealed 2 masses in the pancreas and multiple liver metastases. The liver biopsy confirmed the presence of non-beta islet cell tumors. Okay, tumor tissue. The patient is most likely to be suffering from which of the following diseases? Write your answers in the chat. Perfect. Okay, this makes me so happy that you guys are answering correct. Okay, the correct answer here is Zollinger-Ellison syndrome. So how will you tackle the question? Okay, I would highlight the fact that he's a 44-year-old man. Okay, but all of this, like mainly here, what's important is that an enlarged pancreatic head was identified. Okay, and ulcers in the duodenum occurred. Why? Because of increased, like severely increased gastrin levels, and we know that gastrin releases acid, okay, causing ulcers. Okay, this revealed multiple masses in the pancreas and liver. We know that gastrinomas mainly occur in the pancreas. This is in Zollinger-Ellison disease, or Zollinger-Ellison syndrome. We know that it mainly affects the pancreas, which is a tumor in the pancreas. That's why there's masses in the pancreas. Okay, and we know that it's not the liver biopsies confirm the presence of non-beta islet cell tumor disease. Okay, this isn't what you're going to get. The main part you're going to focus on is the pancreas. Okay, and the gastrin. This is why the answer is Zollinger-Ellison syndrome. Okay, clear? Do I move on to the next question? Okay, so a 48-year-old man with a recurrent history of ulcer disease associated with diarrhea and a strong family history of duodenal ulcer disease is suspected of having Zollinger-Ellison syndrome, okay? Also known as gastronoma, okay? Secretin was given as rapid intravenous injection to test for gastronoma. Which of the following results supports the existence of gastronoma after secretin administration? Write our answer in the chat. This might be, I feel like the way that this question is written is supposed to like confuse you, but I really wanted to include it because this type of question is really like common to get you to like be in your exam. And I want you guys to think about this question because I mentioned this in the lecture. So what's your answer? Okay, perfect. You guys are making me so happy. Okay, so the answer to this question is B. Okay, why is it B? Let's like digest the question together so you guys know the answer. Okay, basically this is saying that there's this patient who has an ulcer. Okay, with diarrhea. Okay, we know that. Okay, and sorry, and this patient is suspected to have Zollinger-Ellison syndrome. Okay, which is a gastronoma. Now we know that Zollinger-Ellison syndrome gives us duodenal ulcers. Check, okay? But what else do we know? There's another hint in this question, okay? We just said that Zollinger-Ellison syndrome causes staturia. Remember, because Zollinger-Ellison syndrome is a gastronoma in the pancreas, leading to decreased pancreatic enzymes, okay? So lipase won't be secreted, causing staturia. And here they mentioned that this patient has diarrhea. So not only does the common presentation show, which is duodenal ulcer disease, but they also gave you an additional hint that this patient has diarrhea, okay? What's another hint? That when the patient was given secretin, which we know that if you, in Zollinger-Ellison syndrome, okay, if you give secretin, the patient's gastrin won't decrease, even though secretin is a feedback inhibitor of gastrin, but in this case, gastrin isn't secreted by the G cells and secretin acts on the G cells. Here, it's through a gastronoma, tumor. That's why gastrin levels didn't decrease. So what's the result? Even though secretin was administered, the secretin won't inhibit the won't decrease the levels of gastrin. So we will still have increased serum gastrin, okay? Because the source of gastrin here is the tumor, not G cells. Clear? Okay, I'll move on. I'm really happy you guys are answering them so far. Okay. This question is a 44-year-old woman with hematemesis, okay, has heartburn and stomach pain. Endoscopy shows inflammation involving the gastric body and antrum, as well as gastric ulcer, a small gastric ulcer. Biopsies were positive for H. pylori, which damages the gastric mucosa, primarily by increasing mucosal levels of which substance? Write your answer. Okay, yay, okay, you guys are answering it right, okay? Perfect, perfect. Good job, everyone. Okay, so the answer here is A. Let's digest the question, okay? This patient presents with hematemesis, okay, which is like throwing up blood, okay? And heartburn, okay? We know that. Why heartburn? Maybe because she has GERD, okay? And stomach pain, peptic ulcer, okay, that's an indication, okay? Endoscopy shows inflammation involving the gastric body and antrum, okay? We just said that H. pylori mainly infects the antrum. Do you guys remember? Okay. As well as small gastric ulcers. H. pylori also causes small gastric ulcers. And they gave us the diagnosis of H. pylori, okay? H. pylori damages the gastric mucosa. How? By increasing ammonium levels, okay? Do you guys remember all the mechanism of H. pylori? This is basically this slide that I said is very important, okay? This one, okay? When urea goes into your into H. pylori through urease, it will give us ammonia and bicarb and carbon dioxide, sorry, and it will form this ammonia cloud to evade the acid, which kills the bacteria, but it will evade it, so it will not get killed. Okay, that's the answer to the question. Okay, question clear? Should I move on? Okay, so what would you find in a person with Zollinger syndrome? Okay, by the way, I got this question from 13th batch past paper. That's my batch, okay? But only the correct answer was in it's written in the past paper, so I included the rest of the answers like from, I added other options. Perfect. Okay, Satyria. This is because as I just explained, the what is it called? One of you answered A. Okay, so the correct answer is B, which is Satyria. Okay. Zollinger-Ellison syndrome is mainly associated with Satyria. Okay. Why? Because we just said that Zollinger-Ellison syndrome is a gastronoma, which is a tumor in the pancreas. Okay, leading to decreased pancreatic enzymes, which in turn, lipase will decrease since lipase is a pancreatic enzyme, so it will not break down lipid. Okay, so it will cause satyria. Okay, which is like lipidy, like fatty diarrhea. That's what like satyria means. Okay, is that answer like clear? To the person that answered A, you don't have your name, but is it clear why the answer is B, not A? Okay, perfect. So this question is also from 13th batch past paper, and I added the other options even though they only included like the correct option. Okay, so what factors cause gastric mucosal defect? Good job, Salma. Good job, Khalid. Good job, the person that doesn't have their name written. Okay, perfect. Okay, so the answer is C. Okay. Yeah, so gastric mucosal defect can be caused by decreased mucosal blood flow. So why is the answer not A? Okay, because increased mucus doesn't defect the gastric mucosa. Okay, it's a protective mechanism. Okay, the same with increased mucosal blood flow. The same with decreased HCl. So the only correct answer here is C. Okay, which is decreased mucosal blood flow. Okay. So thank you everyone so much for listening. Okay, if you have any questions, please don't hesitate to ask me on my either WhatsApp or like on email. On the last slide here, okay, I added like useful resources, okay, that I found, okay, either on Ambos or Dr. Peter, whatever. I really recommend you guys to read it. Okay, so Dr. Peter's book, okay, which is Physiology and Pathophysiology of Digestion. You can find it in the resource slide, okay, and the resource folder. I think you guys have access to the resource folder. Read from page 125 to 133. It explains probably its entire lecture, okay? Ambos. Any question you have, you can Google it on Ambos. I also included a lot of slides from Ambos, so read through them, okay? This video, really recommend you to read it, to watch it. I watched this video to help me explain it to you guys. It's so helpful. It's about the urea breath test, like the lady explains it really well, okay? This is a video about gastric mucosal defense. It basically summarizes gastric mucosal defense. And this is the only 2-minute summary of gastric mucosal defense. I also really recommend you guys to read it. And this video is Physiology of Vomiting. This is the same one I included in that slide. So I really recommend you guys to go through all these resources and don't hesitate to like ask me any questions, like my email or in my WhatsApp. And I'm free now for any questions. Khalid asked, can you summarize the urea test? Okay, yes. So going back to the slide, oh, by the way, for everyone that doesn't have a question, feel free to leave, okay? And anyone who does have a question, like class the session ended, like I will explain it, like I can answer any questions you guys have now, okay? Thank you guys so much for listening. Inshallah, this helped. Okay. You're welcome, guys.